Right now, beneath the frozen tundra of Siberia, something ancient is stirring. Deep in the permafrost, locked in ice that has remained solid for tens of thousands of years, viruses that infected organisms when mammoths walked the earth are waiting. They are not dead, they are dormant. And as the Arctic warms at a pace 4 times faster than the rest of the planet, these frozen time capsules are beginning to thaw. In 2014, a team of scientists led by Jean Michel-Claverie, an emeritus professor of genomics at Aix-Marseille University in France, accomplished something that sounds like science fiction. They took a sample of Siberian permafrost that had been frozen for 30,000 years, brought it to their laboratory, and deliberately woke up a virus that had been inactive since the last ice age. The virus, which they named Pithavirus sibricum, was extracted from coastal tundra approximately 30 meters below the surface. When they introduced this ancient pathogen to cultured amoeba cells, the virus sprang to life. It infected the amoebae with the same efficiency as any modern virus, replicating itself and bursting from host cells to infect new targets. 30,000 years of frozen stasis had not diminished its ability to infect. The experiment proved something profound. Viruses can survive, fully viable and infectious, for geological time spans. This was not a one-time fluke. Claverie and his team repeated the process in 2015 with a different virus type, achieving identical results. But their most remarkable work came in 2023, when they published findings that pushed the boundaries even further. Working with 7 different permafrost samples collected from various locations across Siberia, the researchers isolated multiple strains of ancient viruses and demonstrated that each could still infect amoeba hosts. These samples represented different time periods and environments. Some came from the stomach contents of a frozen woolly mammoth, dated to approximately 27,000 years ago. Others were extracted from deeper, older layers. The oldest strain came from a sample taken from an underground lake 16 meters beneath the surface. Radiocarbon dating of the surrounding soil revealed an age that seemed almost impossible, 48,500 years. Think about that timeline. 48,500 years ago, Neanderthals still walked Europe and Asia. Modern humans were just beginning to spread across the continents. The last ice age was approaching its peak, and in the Siberian soil, a virus infected an amoeba, then became entombed in freezing sediment. That virus remained frozen through the entirety of human civilization. It was frozen when the first cities rose in Mesopotamia. It was frozen when the pyramids were built, when Rome fell, when the Americas were colonized, when the Industrial Revolution transformed the world. And when scientists thawed it in their laboratory in France, it resumed its infection cycle as if no time had passed at all. The term Claverie uses for these ancient pathogens is deliberately evocative: zombie viruses. The name captures both the scientific reality and the unsettling implications. These viruses are not resurrected in the mystical sense. They never truly died. Instead, they entered a state of suspended animation so complete that their molecular machinery remained perfectly preserved. The virus particles retained their structural integrity. Their DNA genomes remained intact. The proteins forming their capsids, the protective shells surrounding their genetic material, maintained their functional shapes. When conditions changed, when warmth and moisture returned, these viral time travelers simply picked up where they left off. The process raises immediate questions. How is such preservation possible? What are the mechanisms that allow complex biological molecules to remain stable for tens of thousands of years? The answers lie in the unique properties of permafrost itself. Permafrost is ground that remains frozen year-round for at least 2 consecutive years, though most Arctic permafrost has been frozen for far longer. Some regions contain ice that has remained solid for hundreds of thousands of years. The uppermost layer, called the active layer, thaws during summer and refreezes in winter, but below that seasonal boundary, the ground stays perpetually frozen. In Siberia, permafrost can extend hundreds of meters deep. The temperatures remain well below freezing, typically ranging from -5 to -15 degrees Celsius, though some areas are colder still. These frigid conditions slow molecular degradation to an almost imperceptible crawl. A But cold alone does not explain the remarkable preservation. Permafrost also provides an oxygen-free environment. When soil freezes solid, air pockets are sealed off, and oxygen is largely excluded. Without oxygen, the chemical reactions that normally break down organic molecules cannot proceed efficiently. Oxidation, one of the primary mechanisms of molecular decay, essentially stops. Additionally, permafrost is completely dark. Ultraviolet radiation, which damages DNA and proteins, cannot penetrate the frozen soil. The combination creates a perfect preservation medium, cold, anoxic, and shielded from radiation. Within this frozen matrix, viral particles settle into a state that biologists describe as being on the edge between living and non-living. Viruses occupy a strange position in biology. Outside a host cell, a virus is essentially an inert package of genetic information wrapped in a protein shell. It does not metabolize, it does not grow, it does not respond to stimuli. By most definitions of life, a virus particle floating in water or frozen in ice is not alive. But introduce that same virus particle to an appropriate host cell and everything changes. The virus penetrates the cell, hijacks its machinery, and transforms it into a factory for producing more viruses. During this infection cycle, the virus cell system does exhibit the characteristics of life. It grows, reproduces, and responds to its environment. In permafrost, viruses wait in this liminal state. Their DNA, the blueprint for infection, remains chemically stable because DNA molecules, while complex, are relatively resistant to degradation when protected from heat, water, oxygen, and radiation. The double helix structure provides some inherent stability, with each strand backing up the information in the other. Even if some damage occurs over millennia, certain viral genomes are large enough that they can tolerate a degree of mutation and still function when thawed. The protein capsids surrounding the genetic material also remain remarkably stable at frozen temperatures. Proteins can denature, losing their functional shape, but this process requires energy, typically in the form of heat. At permafrost temperatures, proteins remain locked in their folded configurations, essentially indefinitely. The research by Clavery's team focused specifically on a category of viruses called giant viruses, which we will explore in depth in the next chapter. These viruses are unusually large, visible under standard light microscopes, making them ideal subjects for this type of research. But the implications extend far beyond this single virus family. When scientists examine permafrost samples using genetic sequencing techniques, they find evidence of staggering viral diversity. Not just dozens or hundreds of different virus types, but potentially tens of thousands of distinct viral species preserved in the frozen soil. The Siberian permafrost, stretching across millions of square kilometers, represents an archive of ancient microbial life unmatched anywhere else on Earth. As organisms died and were incorporated into accumulating sediments that subsequently froze, their associated viruses were entombed alongside them. Layer upon layer, year after year, millennia after millennia, this biological library grew. Scientists have only begun to sample this archive. Every new core sample reveals viral genomes never seen before, genetic sequences with no match in existing databases, evidence of extinct viral lineages that once thrived in ecosystems now vanished. But this frozen archive is not stable. The Arctic is warming. Average temperatures in the region have increased approximately 3 to 4 degrees Celsius since the mid-20th century, and the rate of warming continues to accelerate. This temperature rise is thawing permafrost that has remained frozen since the Pleistocene. The active layer, the surface zone that thaws each summer, is growing deeper. In some regions, permanently frozen ground is thawing for the first time in 50,000 years or more. The implications are profound on multiple levels. As permafrost thaws, organic matter that has been frozen becomes available for microbial decomposition. Bacteria and fungi begin breaking down plant material, animal remains, and other organic compounds that have been preserved in ice. This decomposition releases greenhouse gases, primarily carbon dioxide and methane. The permafrost regions of the Arctic contain an estimated 1,600 billion metric tons of carbon, nearly twice the amount currently in Earth's atmosphere. As this carbon is released, it accelerates global warming, which causes more permafrost to thaw, creating a self-reinforcing feedback loop. Within this thawing landscape, the zombie viruses are awakening, not metaphorically, but literally. As ice melts and frozen soil becomes liquid, viral particles that have been immobilized for millennia are now free to move. If their host organisms are present, infection can occur. The research proving this is not theoretical. Claverie and his colleagues have demonstrated it in controlled laboratory conditions. The viruses they studied specifically infect amoebae, single-celled organisms common in soil and water. These amoebae exist in thawing permafrost regions. When virus meets host, infection proceeds. The zombie virus experiments were conducted under strict safety protocols. Claverie deliberately chose to work with viruses that infect only amoebae, not animals or humans. Every revived virus was tested to confirm it could not infect human or animal cell lines. This precaution was essential because the experiment opened a door to an uncomfortable question. If amoeba-infecting viruses remain viable after 48,000 years in permafrost, what about viruses that infect other organisms? Permafrost does not exclusively preserve harmless microbes. It also contains the remains of animals and humans, along with whatever pathogens they carried. Anthrax bacteria, which form extremely durable spores, have already been proven to survive in frozen ground and cause outbreaks when thawed. In 2016, a heat wave in Siberia thawed a reindeer carcass that had been frozen for decades. The carcass still harbored viable anthrax spores. When those spores were released into the environment, they caused an outbreak that killed more than 2,000 reindeer and hospitalized dozens of people, with one child dying from the infection. If anthrax can survive and remain dangerous, the question becomes unavoidable. Can ancient viruses that infect mammals, including humans, also survive? The scientific consensus is that the risk of a pandemic from permafrost viruses is low, but not zero. Virologists emphasize that most viruses are highly specific to their hosts, and that jumping to a new host species is difficult and rare. Additionally, human immune systems have evolved in a world full of viruses, developing broad-based defenses that can respond to novel pathogens. A virus that last infected humans or their ancestors tens of thousands of years ago would encounter a very different immunological landscape than the one it originally navigated. However, the possibility cannot be entirely dismissed. Smallpox, which has been eradicated in the wild, is theoretically present in human remains buried in permafrost. If viable smallpox virus were released from thawing ground in a region where vaccination has ceased, the consequences could be severe. Claverie himself has spoken about these risks in interviews. He emphasizes that the zombie virus experiments serve as a proof of concept. They demonstrate that ancient viruses can remain infectious, which implies that monitoring and surveillance of thawing permafrost regions should be a public health priority. The experiments also reveal the sheer diversity of viral life preserved in ice. With every sample tested, new virus families are discovered. The 70,000 species mentioned in this script's title is not hyperbole. It is a conservative estimate based on metagenomic surveys that we will explore in later chapters. We stand at a unique moment in planetary history. For the first time in tens of thousands of years, the Arctic permafrost is undergoing rapid, widespread thawing, and for the first time ever, humans have the scientific tools to study what emerges from that ice. The frozen time capsule is opening, revealing a hidden world of ancient viruses, some familiar, others utterly alien. What we find in that ice will reshape our understanding of virology, evolution, and the history of life itself. Chapter 2: What makes a virus giant? For more than a century, virologists operated under clear definition of what a virus was. Viruses were tiny, far smaller than bacteria, so small they could not be seen with a light microscope. They were simple, containing minimal genetic material, just enough to hijack a host cell and replicate. They were obligate parasites, utterly dependent on cells for every aspect of their reproduction. This definition held firm through decades of research. Viruses were the ultimate biological minimalists, stripped-down genetic pirates operating at the lower boundary of biological complexity. Then, in 2003, scientists discovered something that shattered every assumption. They found a virus so large it had been mistaken for a bacterium, so complex it contained more genes than some living organisms, so different from all known viruses that it forced a complete rethinking of what viruses could be. The discovery happened in a cooling tower in Bradford, England, though the true significance was not recognized for years. In 1992, Timothy Robotham, a microbiologist at Leeds Public Health Laboratory, was investigating a local pneumonia outbreak. He collected water samples from a power plant cooling tower, a known reservoir for bacterial pathogens. When he examined the samples under his microscope, he identified what appeared to be a bacterium associated with amoebae in the water. He labeled it a Bradfordcoccus, assuming it was a bacterial species. The sample was stored, and the investigation moved on. More than a decade later, researchers at the Mediterranean University in Marseille, France, were studying microbes that infect amoebae. They obtained the Bradford sample and began culturing it, but when they tried to analyze the presumed bacterium using standard bacterial identification techniques, nothing worked. The organism did not respond to antibiotics. Its genetic material did not match bacterial sequences. Detailed electron microscopy revealed that what everyone had assumed was a bacterium was actually something entirely different. It was a virus, but not any virus known to science. The virus particles were enormous. Each measured approximately 750 nanometers in diameter, making them larger than some small bacteria. To put this in perspective, a typical influenza virus is about 100 nanometers across. The common cold virus is roughly 30 nanometers. Most viruses range from 20 to 300 nanometers. This thing was 750 nanometers. It was not just larger, it existed on a completely different scale. In fact, it was large enough to be visible under a standard light microscope, something no one believed was possible for a virus. But size was just the beginning. When the researchers sequenced its genome, they found it contained 1.2 million base pairs of DNA. Most viruses have genomes ranging from a few thousand to a few hundred thousand base pairs. RNA viruses like influenza or coronaviruses typically have genomes of 10,000 to 30,000 bases. Even large DNA viruses, like the herpes virus that causes chicken pox, have genomes of around 150,000 base pairs. This newly discovered virus had a genome nearly 10 times larger than that. Its genome encoded 979 proteins, 33 non-coding RNA molecules, and 6 transfer RNAs. Some bacteria, which are living organisms with metabolisms and the ability to reproduce independently, have fewer genes than this virus. The discovery team, led by Didier Rayerolt and Bernard Lascola, named it mimivirus, short for mimicking microbe, reflecting how it had been mistaken for a bacterium. The publication announcing mimivirus in 2003 sent shockwaves through the virology community. It was not just a large virus, it was a virus that challenged fundamental assumptions about the viral world. Under an electron microscope, mimivirus revealed a structure of stunning complexity. The viral particle, called a virion, consisted of an icosahedral capsid, a 20-sided protein shell with the geometry of a geodesic dome. This capsid measured about 400 nanometers across and was constructed from a repeating pattern of protein subunits called hexans and pentans. The capsid symmetry was calculated to be between T equals 972 and T equals 1200, where T represents the triangulation number, a measure of geometric complexity. To understand what this means, consider that a typical adenovirus, a relatively complex virus that causes respiratory infections, has a triangulation number of 25. Mimivirus had a capsid 40 times more complex. Protruding from the capsid surface were densely packed protein fibers, each approximately 125 nanometers long, giving the entire virus particle a diameter of around 750 nanometers, including these fibers. These fibers are thought to play a role in attaching to host cells. Inside the capsid was a dense core containing the viral DNA genome. Surrounding this core was an internal lipid membrane, creating a compartmentalized structure reminiscent of cellular organization. The genome itself was even more surprising than the structure. When researchers analyzed the genes encoded in that 1.2 million base pair genome, they found things no virus should have. Mimivirus carries genes for amino acid metabolism, DNA repair, and portions of the protein synthesis machinery. Specifically, it encodes 4 aminosyl tRNA synthetases, enzymes that are fundamental to translation, the process by which cells read genetic information and build proteins. These enzymes attach specific amino acids to their corresponding transfer RNA molecules, a critical step in protein synthesis. In all known cellular life, from bacteria to humans, aminosyl tRNA synthetases are essential. But viruses do not make proteins directly. They hijack the host cell's machinery to do it for them. Why would a virus encode its own translation components? The 4 synthetases found in mimivirus are specific for arginine, cysteine, methionine, and tyrosine. Functional studies confirmed that these viral enzymes work. The methionine tRNA synthetase specifically charges methionine onto its cognate tRNA. The tyrosyl tRNA synthetase does the same for tyrosine, following the standard identity rules for tRNA tyrosine recognition found in archaea and eukaryotes. The virus was not carrying these genes as genetic junk. They were active, functional components of the viral infection strategy. Mimivirus also encodes multiple DNA repair enzymes, including components of the base excision repair pathway, which fixes damaged DNA bases. It has genes for DNA recombination, for manipulating chromatin structure, for regulating gene expression. Some of these genes are more similar to eukaryotic genes than to bacterial or viral genes, suggesting the virus acquired them from its hosts over evolutionary time. Others appear to be ancient, possibly inherited from a distant viral ancestor. The discovery of mimivirus opened the floodgates. Once scientists knew what to look for, giant viruses started appearing everywhere. In 2009, researchers described mammavirus, a close relative of mimivirus isolated from the same cooling tower water in Paris. Mammavirus had a similar size and genetic complexity, but with one additional surprise. It was infected by an even smaller virus, which we will discuss in detail in the next chapter. Then came the truly enormous viruses. In 2013, scientists announced the discovery of Pandoravirus, isolated from coastal sediments off Chile and from a pond in Australia. Pandoravirus particles were shaped like amphoras, ancient Greek storage jars, measuring up to 1 micrometer in length and half a micrometer in diameter. 1 micrometer is 1,000 nanometers, making these viruses more than 10 times larger than a typical bacterium. Their genomes were correspondingly massive, 2.5 million base pairs, encoding an estimated 2,500 proteins. Approximately 93% of Pandoravirus genes had no similarity to any known genes in databases, suggesting these viruses represented a completely novel lineage. The same year Pandoravirus was described, Clavery's team, the same researchers who would later revive 48,000-year-old viruses, published their discovery of Pithavirus cybricum from Siberian permafrost. Pithavirus was even larger than Pandoravirus in terms of physical size. The virion measured 1.5 micrometers in length and half a micrometer in diameter, making it the largest virus particle known at the time. It was large enough that under certain microscopy conditions, individual virus particles were visible as tiny specks. Interestingly, despite its enormous size, Pithavirus had a smaller genome than Pandoravirus, containing approximately 610,000 base pairs encoding around 470 proteins. This raised a fascinating question. If genome size does not determine virion size, what does? Many viruses pack their genomes extremely efficiently, cramming DNA tightly into compact capsids. Pithavirus, by contrast, had a spacious interior. The reason remains unclear, but it may relate to how the virus replicates inside host cells. Unlike many giant viruses, Pithavirus does not encode any components of the protein translation machinery, such as aminosyl tRNA synthetases. However, it does encode a complete set of machinery for producing messenger RNAs, including enzymes for transcription. These proteins are packaged inside the virus particle, ready to function immediately upon infection. Pithevirus carries out its entire replication cycle in the cytoplasm of the host cell, much like poxviruses, the family that includes smallpox. The virus apparently brings everything it needs to transcribe its genes and does not rely on entering the host nucleus. In 2018, researchers described Tupinvirus, isolated from sewage and lake sediments in Brazil. Tupinvirus belongs to the same family as Mimivirus, but has a unique feature, a long-tail structure extending from one end of the icosahedral capsid, resembling a bacteriophage. The genome is approximately 1.5 million base pairs, encoding around 1,400 proteins. Remarkably, Tupinvirus encodes the most complete set of translation machinery ever found in a virus. It carries genes for 20 aminosyl tRNA synthetases, covering all 20 standard amino acids used in proteins, along with 70 transfer RNAs and numerous translation factors. This virus has essentially assembled a nearly complete protein synthesis system, though it still depends on the host ribosome to actually build proteins. All these giant viruses belong to a larger group called the nucleocytoviricota, previously known as nucleocytoplasmic large DNA viruses, or NCLDV. This group encompasses several families, including Mimiviridae, which includes Mimivirus and its relatives, Pandoraviridae, Pandoravirus, Pithaviridae, Pithavirus, Marsaleviridae, Ascoviridae, Asfarviridae, which includes African swine fever virus, Poxviridae, smallpox and relatives, Phycodnaviridae, viruses infecting algae, and Iridoviridae, viruses infecting insects and cold-blooded vertebrates. Despite their diversity in size, shape, and genome content, these families share certain core genes involved in DNA replication and virion structure, suggesting they all evolved from a common ancestor. The evolutionary origin of giant viruses remains hotly debated. Some researchers propose they evolved from an ancient cellular lineage that gradually lost functions and became parasitic, eventually arriving at the viral lifestyle. This would make giant viruses the remnants of a fourth domain of life, distinct from bacteria, archaea, and eukaryotes. Evidence for this includes the presence of genes found in no other viruses and the sheer genomic complexity, which seems difficult to achieve through gene acquisition alone. Other scientists argue that giant viruses evolved from simpler viruses through extensive gene acquisition from their hosts. According to this model, an ancestral virus, possibly similar to a bacteriophage, infected early eukaryotic cells and began stealing genes. Over millions of years, the virus accumulated hundreds or thousands of genes, becoming progressively larger and more complex. Phylogenetic analyses support this scenario for at least some components of giant virus genomes. Many giant virus genes are clearly related to eukaryotic genes, suggesting horizontal gene transfer from hosts. The truth may lie somewhere between these extremes. Giant viruses likely have a complex evolutionary history involving both an ancient viral ancestor and extensive gene acquisition. Some of their core genes appear genuinely ancient and distinctly viral. Others are obvious acquisitions from eukaryotes, bacteria, or even other viruses. The mosaic nature of giant virus genomes, with genes from multiple sources stitched together, reflects billions of years of genetic piracy and exchange. What is undeniable is that giant viruses blur the line between viruses and living organisms. The traditional definition of a virus as a simple genetic parasite does not adequately describe an entity with over 2,000 genes, many encoding functions associated with metabolism and information processing. Some scientists have proposed a new concept called the virocell, which considers the infected cell as the actual viral organism rather than just the virion particle. Under this view, the virus particle is merely a transmission stage, like a spore or seed, while the actively replicating virus cell system is the living entity. This paradigm shift has profound implications for how we understand viruses and their place in the tree of life. Giant viruses also challenge our understanding of viral ecology. They are not rare oddities. Environmental sampling has revealed they are common in oceans, lakes, soils, and other habitats worldwide. They infect a variety of hosts, including amoebae, algae, and other protists. By infecting and killing these microorganisms, giant viruses influence nutrient cycling, population dynamics, and ecosystem structure in ways we are only beginning to understand. And as we now know from the permafrost research, these giant viruses can persist in a viable state for tens of thousands of years, waiting in frozen soil for conditions to change. The 70,000 species hiding in permafrost include countless giant viruses, representing lineages that may be extinct everywhere else on Earth. Each thawing sample potentially releases viruses the modern world has never encountered, viruses that infected organisms in Pleistocene ecosystems radically different from today. We are only beginning to catalog this diversity and understand what these ancient giants can teach us about the deep history of viral evolution. Chapter 3: The Discovery That Changed Virology. The story of how giant viruses went from a single bizarre anomaly to a recognized viral supergroup spanning the globe is a tale of technological revolution, persistent researchers, and paradigm-shattering discoveries. It is a story that unfolded over 2 decades and fundamentally transformed our understanding of the viral world. When mimivirus was formally described in 2003, many virologists were skeptical. The virus was too large, too complex, too different. Some questioned whether it truly was a virus or whether it represented some kind of highly reduced parasitic bacterium. The defining characteristic of a virus is that it cannot replicate independently. It must hijack a host cell's machinery. Mimivirus fit this criterion. Despite its enormous genome and gene content, it could not reproduce without infecting an amoeba. It encoded many cellular-like genes, but not a complete metabolism. It had aminosyl tRNA synthetases, but no ribosomes, the cellular machines that actually synthesize proteins. It was a virus, just unlike any virus known before. The key insight that allowed the discovery of mimivirus was methodological. Traditional virology relied on filtering techniques to separate viruses from bacteria. Researchers would pass samples through filters with pore sizes of 200 or 300 nanometers, small enough to trap bacteria but large enough to let viruses through. Mimivirus, at 750 nanometers, would have been trapped by these filters and discarded with the bacteria. It was only when researchers examining amoeba-associated microbes used direct microscopy and genetic techniques that did not rely on size filtration that mimivirus was recognized. This realization sparked a search for other large viruses that might have been overlooked. If one giant virus existed, there were probably others. Researchers began examining environmental samples using approaches that did not pre-filter by size. They cultured amoebae in water samples and looked for signs of infection. They used genetic sequencing to survey the viral diversity in sediments, oceans, and soils without assumptions about size or structure. The results were stunning. In 2009, the discovery of mammavirus confirmed that mimivirus was not unique. Mammavirus was isolated from a water sample from a cooling tower in Paris and showed striking similarity to mimivirus in size, structure, and gene content. Its genome was approximately 1.2 million base pairs, encoding around 1,000 proteins. But mammavirus came with a surprise. It was infected by a smaller virus called Sputnik, which replicated inside the mammavirus particle and interfered with its reproduction. This discovery opened an entirely new chapter in virology, the existence of viruses that parasitize other viruses, but that story belongs in the next chapter. In 2010, researchers described megavirus chilensis, isolated from seawater off the coast of Chile. Megavirus had a genome of 1.26 million base pairs, slightly larger than mimivirus, and encoded over 1,000 proteins. It reinforced the emerging picture that giant viruses were not confined to cooling towers or specific environments. They existed in marine ecosystems, freshwater, and terrestrial habitats. The real explosion of diversity came with the advent of metagenomic sequencing. Traditional virology required culturing viruses, growing them in host cells in the laboratory. But the vast majority of viruses cannot be cultured using standard techniques, either because their hosts are difficult to grow or because the right host has not been identified. Metagenomics bypasses this limitation by sequencing all the DNA in an environmental sample without culturing anything. Researchers extract DNA from water, soil, or sediment, sequence it, and then use computational methods to identify which sequences belong to viruses based on their genetic signatures. When scientists applied metagenomic approaches to oceans and lakes, they uncovered extraordinary viral diversity. The Tara Oceans Expedition, a global oceanographic survey, collected water samples from around the world and sequenced the microbial and viral communities in each. The results revealed thousands of previously unknown giant virus sequences. Many belonged to known families like Mimiviridae or Phycodnaviridae, but others represented entirely novel lineages. The ocean, it turned out, was teeming with giant viruses infecting algae, protists, and other microorganisms. Similar surveys in freshwater lakes found abundant giant viruses. Samples from Antarctic lakes, Arctic ponds, tropical rivers, and temperate reservoirs all contain diverse populations of large DNA viruses. Some infected algae playing crucial roles in controlling algal blooms. Others infected amoebae or other protists. The ecological importance of these viruses became clear. By killing their hosts, giant viruses influence nutrient cycling, energy flow, and community structure in aquatic ecosystems. In 2013 came the discovery of Pandoravirus, which reset expectations for just how large a virus could be. Two species were initially described: Pandoravirus salinus from coastal sediments off central Chile and Pandoravirus dulcis from a freshwater pond near Melbourne, Australia. The virions were amphora-shaped, measuring up to 1 micrometer long and half a micrometer wide. To put this in perspective, a typical human red blood cell is about 7 micrometers across, meaning you could line up 7 Pandoravirus particles across a single blood cell. These were not just viruses visible under a light microscope, they were easily visible, appearing as distinct ovoid particles. The genomes were equally staggering. Pandoravirus salinus had a genome of 2.47 million base pairs, while Pandoravirus dulcis had 2.5 million base pairs. These genomes encoded an estimated 2,500 proteins each. But here was the truly shocking part. 93% of the genes in Pandoravirus had no recognizable similarity to any genes in public databases. They were not just new, they were utterly alien, with no clear evolutionary connection to genes in bacteria, archaea, eukaryotes, or other viruses. This suggested Pandoravirus represented a deeply divergent lineage that had been evolving independently for an immense span of time. That same year, 2013, Jean Mitchell-Clavery's team published their discovery of Pithavirus cybricum from 30,000-year-old Siberian permafrost. As discussed in the previous chapter, Pithavirus was the largest virus particle yet discovered, at 1.5 micrometers long, though its genome was smaller than Pandoravirus at 610,000 base pairs. The significance of Pithavirus extended beyond its size. It was the oldest virus ever revived and proven to be infectious, demonstrating that ancient viruses in permafrost were not just preserved genetic sequences, but viable, dangerous pathogens. The discoveries continued. In 2015, researchers isolated Mollivirus cybricum from the same Siberian permafrost sample that yielded Pithavirus. Mollivirus was also a giant virus, with a spherical shape and a genome of approximately 650,000 base pairs. In 2017, Cedritvirus was described from a freshwater sample in France, representing yet another distinct lineage within the giant virus world. In 2021, scientists reported the discovery of viruses in the Gyulya ice cap on the Tibetan plateau. Ice core samples spanning 15,000 years contained genetic signatures of approximately 1,700 different viral species, many of them previously unknown. The majority were temperate bacteriophages, viruses that infect bacteria, but the diversity was remarkable. The study demonstrated that glacial ice, like permafrost, serves as an archive of ancient viral communities, preserving a record of microbial life across millennia. By 2022, metagenomic studies of permafrost were revealing the true extent of viral diversity frozen in Arctic soils. Researchers analyzing samples from multiple sites across Siberia, Alaska, and Canada found genetic evidence for tens of thousands of distinct viral genotypes. Many of these sequences belong to giant virus families, including numerous novel lineages within Mimiviridae and Pithaviridae. Some samples showed giant viruses making up 12% of all DNA sequences, indicating these viruses were dominant members of ancient soil ecosystems. A particularly significant study published in 2022 reported the recovery of a complete 1.6 million base pair genome belonging to the Pithaviridae family from 42,000-year-old permafrost. This genome was fully assembled from metagenomic data without culturing the virus, demonstrating the power of modern sequencing to reconstruct ancient viral genomes. The genome encoded genes for DNA replication, transcription, virion structure, and numerous proteins of unknown function. Phylogenetic analysis placed it as a novel member of the Pithaviridae, distinct from Pithavirus cybricum but clearly related. The geographic distribution of these discoveries was revealing. Giant viruses were being found on every continent and in virtually every aquatic environment sampled. Marine samples from tropical, temperate, and polar oceans all contained diverse giant viruses. Freshwater lakes, rivers, and ponds harbored their own giant virus communities. Soil samples, when analyzed with appropriate techniques, revealed terrestrial giant viruses. Permafrost and glacial ice contained frozen archives of ancient viral diversity. There was no environment too extreme, no location too remote. The diversity extended to viral morphology and genomic content. Some giant viruses, like mimivirus and its relatives, had icosahedral capsids covered in protein fibers. Others, like Pandoravirus, had amphora-shaped particles with an apparently different internal structure. Pithavirus had an ovoid morphology with a distinct cork-like structure sealing one end. Mollivirus was nearly spherical. Tupinvirus had a tail. Each morphology reflected different strategies for entering host cells and organizing the viral genome. Genomic diversity was equally impressive. Genome sizes ranged from around 400,000 base pairs in some Marseilleviruses to over 2.5 million in Pandoravirus. Gene content varied dramatically. Some giant viruses encoded extensive translation machinery, others had none. Some carried genes for photosynthesis-related proteins, apparently stolen from algal hosts. Others had genes involved in lipid metabolism, sugar metabolism, or nutrient transport. The mosaic nature of these genomes reflected extensive horizontal gene transfer, with viruses acquiring genes from hosts, other viruses, and even bacteria. Phylogenetic analyses, attempting to reconstruct the evolutionary relationships among giant viruses, revealed complex histories. Some gene trees showed clear clustering of viruses by family, suggesting vertical inheritance from common ancestors. Other gene trees showed completely different relationships, indicating those particular genes had been acquired independently through horizontal transfer. The evolutionary history of giant viruses was not a simple tree, but a tangled web of inheritance, acquisition, and loss. By 2025, the number of recognized giant virus families had grown substantially. The International Committee on Taxonomy of Viruses, the body responsible for naming and classifying viruses, had expanded the Nucleositoviricota phylum to include more than a dozen distinct families. New metagenome-assembled genomes were being reported monthly, each adding to the known diversity. Conservative estimates suggested at least 70,000 distinct giant virus species existed globally, though the true number could be far higher. What enabled this explosion of discovery was technology. The development of high-throughput DNA sequencing made metagenomic surveys feasible and affordable. The creation of massive genetic databases allowed researchers to compare new sequences against millions of known genes. Advances in microscopy, including cryo-electron microscopy, revealed viral structures in unprecedented detail. Bioinformatics tools for assembling genomes from fragmented metagenomic data improved dramatically. Each technological advance opened new windows into viral diversity. The discovery timeline also reflected changing scientific paradigms. In the 1990s and early 2000s, viruses were still largely viewed through the lens established in the mid-20th century: small, simple, gene-poor parasites. Mimivirus forced a confrontation with that paradigm. Initially dismissed by some as an oddity, it was eventually recognized as representing a fundamental expansion of known viral diversity. Each subsequent discovery reinforced the lesson: viruses are far more diverse, complex, and important than traditional virology assumed. Today, the study of giant viruses is a thriving field intersecting virology, ecology, evolution, and environmental science. Researchers are working to understand how these viruses replicate, how they interact with hosts, how they influence ecosystems, and what their existence tells us about the origins of viruses and even the origins of eukaryotic cells. The ancient viruses preserved in permafrost offer a unique opportunity to study viral evolution across deep time, comparing modern and ancient lineages to understand how viruses change over tens of thousands of years. The story is far from over. As the Arctic continues to warm and more permafrost thaws, new samples become accessible. Each core drilled into frozen ground potentially contains viral genomes from ecosystems that vanished millennia ago. Each metagenomic survey of thawing soil adds thousands of new sequences to databases. The 70,000 species hiding in permafrost represent not an endpoint, but a starting point for understanding the true scope of viral diversity on Earth. The discoveries that changed virology are continuing right now in laboratories around the world as researchers analyze samples from Siberia, Alaska, Greenland, and the Tibetan Plateau. They are finding viruses with genomes larger than any previously known, viruses with gene combinations never seen before, viruses that challenge every remaining assumption about what viruses are and what they can do. The frozen time capsule is yielding its secrets, one thawed sample at a time, and each secret rewrites another chapter in the story of life on Earth. Chapter 4: Virophages, viruses that infect viruses. The discovery of mimivirus in 2003 shattered fundamental assumptions about what viruses could be. But 5 years later, an even stranger discovery would challenge the very concept of viral ecology itself. In 2008, researchers studying mammavirus, a close relative of mimivirus, noticed something peculiar happening inside infected amoeba cells. The giant virus was not replicating normally. Its viral factories, the massive compartments within the cell where new virus particles are assembled, looked malformed. The capsids being produced were asymmetric, abnormal, defective. Production yields were down by 70%. Something was interfering with the virus. When the scientists examined the viral factories more closely using electron microscopy, they found tiny particles, far smaller than mammavirus, clustered inside the factories and apparently replicating there. These small particles measured only 50 nanometers in diameter, making them 1/15 the size of the giant virus. They had icosahedral capsids similar to many other viruses, but their behavior was unprecedented. These particles were not infecting the amoeba cell directly, they were infecting the giant virus itself. The researchers, led by Bernard Lascola and Didier Rayerolt from Aix-Marseille University, isolated the small virus and characterized it. They found it contained a circular double-stranded DNA genome of approximately 18,000 base pairs encoding 21 proteins. The virus could not replicate on its own in amoeba cells. It required the presence of mammavirus. When amoebae were infected with mammavirus alone, the giant virus replicated normally, producing hundreds of new viral particles. When the same amoebae were co-infected with both mammavirus and the small virus, mammavirus replication crashed, and the small virus multiplied explosively, using the giant virus's replication machinery as its own private production facility. This was parasitism on a scale never before documented. Parasites that exploit viruses were unknown. Viruses themselves are parasites, obligate intracellular parasites that cannot reproduce without hijacking cellular machinery. The idea that another virus could parasitize a virus seemed to violate the natural order. Yet here was evidence, clear and reproducible, that this small virus was doing exactly that. It entered cells infected by mammavirus, located the viral factories where mammavirus was replicating, inserted itself into those factories, and commandeered the giant virus's machinery to make copies of itself instead of the giant virus. The discovery team named the virus Sputnik after the first artificial satellite launched by the Soviet Union because it was the first satellite virus ever discovered, one that orbits and depends upon a larger viral entity. But the term satellite virus was not quite right because satellite viruses, while requiring helper viruses for replication, do not necessarily harm their helpers or compete with them for resources. Sputnik was different. It was actively parasitic, reducing mammavirus yields and protecting the amoeba host from being killed as quickly. The researchers coined a new term for this relationship, virophage, meaning virus eater, in analogy to bacteriophages, the viruses that eat bacteria. Sputnik virophage was isolated from the contact lens fluid of a patient with keratitis, an eye infection. The same sample contained both mammavirus and Sputnik, suggesting the two viruses naturally co-occur in environments where amoebae live, which includes water systems, soil, and human-associated environments. Subsequent research confirmed that Sputnik could infect multiple members of the mimivirus family spanning different lineages, designated lineage A, lineage B, and lineage C. This broad host range among related giant viruses suggested Sputnik had evolved sophisticated mechanisms for recognizing and exploiting viral factories. How does a virophage work? The molecular details are still being uncovered, but the basic life cycle is now understood. Sputnik virions attach to the surface of amoeba cells, likely using the same or similar receptors that mimivirus uses. The virophage enters the cell, possibly through endocytosis. Once inside, Sputnik cannot do anything on its own. It lacks the genetic machinery to replicate its genome or transcribe its genes. It must wait for mimivirus or a related giant virus to infect the same cell. When mimivirus infects the amoeba, it establishes a viral factory within the cell's cytoplasm. This factory is a distinct compartment, visible under a microscope, where the giant virus concentrates all the cellular resources it needs for replication. The factory contains enzymes for DNA replication, transcription machinery to make messenger RNA from viral genes, translation components to help synthesize viral proteins, and structural proteins for assembling new capsids. The giant virus essentially creates a semi-autonomous production facility inside the cell. Sputnik detects this factory and migrates toward it, possibly guided by chemical signals or structural changes in the cytoplasm. Once inside the factory, Sputnik releases its genome. The virophage genome is now in the perfect environment, surrounded by DNA replication enzymes, transcription machinery, and protein synthesis components, all provided by the giant virus. Sputnik genes are transcribed using mimivirus machinery. Sputnik DNA is replicated using mimivirus replication enzymes. Sputnik proteins are synthesized using a combination of mimivirus and cellular translation systems. New Sputnik capsids are assembled inside the factory. When the infected cell eventually lyses, releasing its viral cargo, it releases both defective, malformed mimivirus particles and abundant Sputnik virophages. The damage to mimivirus is severe. In controlled experiments, co-infection with Sputnik reduces mammavirus yields by approximately 70%. The viral particles that are produced are often malformed, with capsids that have accumulated asymmetrically on one side of the particle, rendering them non-infectious. Amoeba lysis, the rupture of the cell that normally occurs within 12 hours of mimivirus infection, is delayed threefold when Sputnik is present, extending to 36 hours or more. This delay gives the amoeba more time to mount defenses and potentially survive the infection. From the amoeba's perspective, Sputnik is actually beneficial, a biological control agent that weakens the giant virus. The discovery of Sputnik immediately raised questions. Was this virophage unique, or did other virophages exist? The answer came quickly. In 2010, researchers described Mavirus, a virophage that infects Cafeteria rhombogensis virus, or CRO-V, a giant virus belonging to a different family than Mimivirus. Mavirus was isolated from seawater and showed a different lifestyle than Sputnik. Unlike Sputnik, which exists only as free particles, Mavirus can integrate its genome into the chromosomes of its host protist, Cafeteria burkhardi, a marine heterotrophic flagellate. The integrated Mavirus genome remains dormant until the flagellate is infected by CRO-V. Upon giant virus infection, Mavirus is activated, excises itself from the host chromosome, replicates inside the viral factory, and produces new virophage particles. This integrated virophage provides the flagellate with immunity against the giant virus, converting a lethal infection into a manageable one. The ability of Mavirus to integrate into host genomes is significant. It suggests that virophages may act as a form of adaptive immunity for protists, similar to how CRISPR systems protect bacteria from bacteriophages. The integrated virophage is inherited by daughter cells when the protist divides, providing long-term protection across generations. Researchers examining Cafeteria genomes found that some isolates naturally carried integrated Mavirus, while others did not. Those carrying the integrated virophage were resistant to CRO-V infection. Those without it were susceptible. The same year Mavirus was described, another virophage named Organic Lake Virophage, or OLV, was discovered in Organic Lake, a hypersaline meromictic lake in Antarctica. OLV was found in association with Organic Lake Phycodenavirus, or OLPV, a giant virus that infects algae in the lake. The extreme environment of Organic Lake, with water temperatures below freezing and salt concentrations over 10 times that of seawater, demonstrated that virophages existed not only in typical aquatic environments, but also in the harshest conditions on Earth. As metagenomic sequencing became more widespread, virophage diversity exploded. Researchers began finding virophage-related sequences in environmental DNA samples from oceans, lakes, rivers, soils, and permafrost. A 2013 study analyzing metagenomic data from public databases identified virophage sequences in samples from around the globe. The majority of hits, representing 95% of virophage sequences, were related to OLV, suggesting this virophage lineage is extremely abundant in marine and freshwater environments. Sputnik-related and Mavirus-related sequences were also found, but in lower abundance. The global distribution suggested virophages are common wherever giant viruses exist. This makes ecological sense. If giant viruses are abundant and ecologically important, then parasites of those viruses would also proliferate. The relationship is analogous to predator-prey dynamics in animal ecology. Giant viruses are predators of protists. Virophages are predators of giant viruses. The three-level interaction creates complex population dynamics. By 2015, several additional virophages had been characterized, including Xamalan virophage. Isolated along with its giant virus host from a water treatment plant, Xamalan showed a narrower host range than Sputnik. While Sputnik could infect giant viruses from lineages A, B, and C of the Mimivirus family, Xamalan could only infect lineages B and C. The resistance of lineage of viruses to Xamalan was traced to a defense system called MIMIVII, a cluster of genes that specifically blocks Xamalan replication. This discovery revealed that giant viruses have evolved immune-like systems to defend against virophages, paralleling the immune systems that cellular organisms use to defend against viruses. The existence of antivirophage defenses in giant viruses implies an evolutionary arms race has been ongoing for millions of years. Giant viruses evolve defenses, virophages evolve counter-defenses. Evidence for this arms race comes from genomic analyses showing rapid evolution of genes involved in virophage recognition and defense in giant viruses, and corresponding rapid evolution of genes involved in evading those defenses in virophages. Some virophage genes show clear signs of positive selection, the signature of adaptive evolution driven by selective pressure, in this case, the pressure to overcome giant virus immune systems. A particularly revealing study published in 2020 demonstrated that virophages can expand their host range through mutation. Researchers created a deletion mutant of Guarani virophage, removing a specific gene, and found that this mutant could now infect giant viruses that the wild-type virophage could not. The deleted gene apparently encoded a protein that was recognized by certain giant virus defense systems. By losing that gene, the virophage lost its Achilles heel and gained the ability to infect new hosts. This experimental evolution demonstrated the plasticity of virophage genomes and how quickly they can adapt to new giant virus defenses. In 2018, scientists described Tupin virus, a giant virus with a long-tail structure, and tested whether known virophages could infect it. Guarani virophage, which has a very broad host range among mimivirus family giant viruses, was found to infect Tupin virus and reduce its replication to undetectable levels. The virophage essentially sterilized the giant virus infection, completely blocking virus production. This potent inhibition suggests that under natural conditions, virophages could regulate giant virus populations and prevent them from becoming too dominant in ecosystems. The ecological implications are profound. Giant viruses influence protist populations, controlling algal blooms and regulating nutrient cycling in aquatic systems. Virophages influence giant virus populations, preventing them from eliminating their protist hosts entirely. This creates a three-way interaction: protists, giant viruses, and virophages, each constraining the others in a dynamic balance. Mathematical models of these interactions predict complex dynamics, including oscillations, where protist populations boom, followed by giant virus blooms that crash the protists, followed by virophage blooms that crash the giant viruses, allowing protists to recover. The evolutionary origins of virophages remain debated. Phylogenetic analyses show that virophages share some genes with giant viruses, suggesting they may have originated from giant viruses that lost most of their genes and became specialized parasites of their former relatives. Other virophage genes resemble those found in mobile genetic elements called mavericks or pollentins, which are widespread in eukaryotic genomes. These elements can move between chromosomes and may have given rise to virophages through a process where they escaped the genome and became infectious particles. A particularly intriguing hypothesis is the virophage first scenario, which proposes that virophages are ancient, possibly predating the origin of eukaryotic cells. According to this model, the evolutionary interaction between giant viruses and virophages stretches back over 1 billion years. The billion-year arms race has driven the expansion of giant virus genomes, as these viruses acquired more genes to defend against virophages and to support replication in environments where cellular machinery might be blocked. A 2023 study examining gene phylogenies across virophages and giant viruses found evidence supporting this long co-evolutionary history. One of the most surprising findings is that virophages may have influenced the evolution of giant viruses in fundamental ways. The presence of virophages creates selective pressure for giant viruses to compartmentalize their replication, creating specialized factories where they can concentrate resources and potentially exclude virophages. This compartmentalization may have been a key step in the evolution of the enormous complexity seen in giant viruses today. The viral factories created by mimivirus and its relatives are essentially protoorganelles, membraneless compartments that function like a nucleus within the cytoplasm. Some researchers speculate that the evolutionary pressure from virophages drove giant viruses to become more cell-like, blurring the boundary between viruses and living organisms. As of 2026, dozens of virophages have been identified through culture-based studies and hundreds more through metagenomic surveys. They represent a hidden layer of biological complexity, parasites of parasites that influence ecosystem dynamics in ways we are only beginning to understand. And they exist not only in modern environments, but also in permafrost, frozen alongside the giant viruses they parasitize. When ancient giant viruses are revived from 48,000-year-old ice, their virophages may be revived as well, resuming their billion-year-old conflict as if no time had passed. The zombie viruses awakening from the Arctic permafrost include not just the giants, but also the tiny predators that hunt them, adding another layer to the extraordinary microbial diversity emerging from the thawing ice. Chapter 5: The 70,000 Species Hidden in Ice. When scientists drill into permafrost and extract cores of frozen soil, they are sampling entire ecosystems preserved in time. Each layer of ice contains microorganisms from the era when that sediment was deposited and subsequently frozen. The microbes within represent communities that thrived under climatic conditions vastly different from today. Some cores span tens of thousands of years, creating vertical timelines of microbial life stretching from the present deep into the Pleistocene. And within this frozen archive, viral diversity is staggering. The 70,000 species figure is not hyperbole. It is a conservative estimate derived from metagenomic surveys of permafrost and glacial ice conducted over the past decade. These surveys use high-throughput DNA sequencing to read all the genetic material in a sample without culturing individual organisms. The sequences are then compared against databases of known genes and viruses to identify what organisms are present and estimate how many distinct species exist based on genetic divergence. One of the most comprehensive studies was published in 2022 by researchers examining permafrost samples from Siberia. They collected cores from the Ukhetye-Alas region, an area where permafrost extends hundreds of meters deep and includes layers frozen for over 49,000 years. Surface samples were also taken from Kamchatka, where permafrost is thinner but still ancient. The samples were processed in cleanroom conditions to prevent contamination, and DNA was extracted from each layer. This DNA represented all organisms present in that layer, living or dead, cellular or viral. The sequencing produced billions of base pairs of sequence data. Computational algorithms assembled these short sequences into longer contiguous stretches called scaffolds. These scaffolds were then analyzed to identify which belonged to viruses based on characteristic viral genes, like those encoding major capsid proteins, DNA polymerases, and other hallmark viral machinery. The results revealed extraordinary viral diversity. From the 11 permafrost samples analyzed, the researchers identified 721 scaffolds belonging to the Nucleositoviricota phylum, representing giant viruses. These scaffolds totaled 72 megabases of viral sequence, roughly equivalent to 72 million base pairs. What made the findings remarkable was the heterogeneity. Some samples contained almost no giant virus sequences. Others were dominated by them. In one sample, designated Sample R, giant viruses accounted for an estimated 12% of all DNA sequences when normalized for genome coverage. 12% is an astonishing proportion. It means that for every 8 cellular genomes in the sample, there was roughly one giant virus genome. The sample came from core permafrost beneath a drained thermokarst lake, frozen for over 42,000 years. The high abundance of giant viruses in that ancient soil suggests these viruses were dominant members of the microbial ecosystem at the time the sediment was deposited. The phylogenetic analysis of the viral sequences revealed which families were most abundant. Pithaviridae and Orpheoviridae-like viruses were the dominant groups, followed by Megamimiviridae. Smaller numbers of scaffolds matched Closnuviridae, Phycodnaviridae, and Asfaviridae. The diversity within each family was striking. Comparing the permafrost sequences to known giant viruses showed that most permafrost viral genotypes had never been seen before. They were novel, undescribed, representing lineages that may no longer exist in modern environments or that simply have not been sampled elsewhere. One of the most significant findings was the assembly of a complete giant virus genome from the permafrost metagenome. From Sample R, the 42,000-year-old core, researchers assembled a circular genome of 1.6 million base pairs belonging to the Pithaviridae family. The genome was complete, meaning the entire genetic blueprint of the virus could be reconstructed from the fragmented metagenomic data. This was a technical triumph. Assembling a genome from environmental samples is challenging because the DNA is degraded into short fragments, often just a few hundred base pairs long, and mixed with DNA from hundreds of other organisms. Sophisticated algorithms must piece these fragments together like a jigsaw puzzle, using overlapping sequences to infer the original genome structure. The reconstructed Pithaviridae genome encoded approximately 500 proteins. Many were homologous to genes in Pithavirus cybricum, the virus revived by Clavery's team from 30,000-year-old Permafrost, but there were significant differences. The Permafrost virus represented a distinct lineage, a separate species within the same family. Phylogenetic trees placed it as a sister group to known pithaviruses, suggesting it diverged from them millions of years ago and has been evolving independently ever since. This genome reconstruction demonstrated that even if a virus cannot be cultured or revived, its complete genetic information can be recovered from permafrost. This opens extraordinary possibilities. Researchers can now study ancient viruses genetically without needing to culture them in the lab. They can examine the genes, infer the virus's structure and life cycle, compare it to modern relatives, and trace its evolutionary history, all from DNA extracted from ice. But the 70,000 species estimate encompasses far more than giant viruses. In 2024, a study of the Giulia ice cap on the Tibetan Plateau expanded our understanding of viral diversity in frozen environments. Researchers drilled more than 1,000 feet into the glacier, extracting ice cores spanning 41,000 years. The cores were sectioned into samples representing 9 distinct time periods, including 3 major climate transitions, shifts from cold glacial conditions to warmer interglacial periods. From these samples, researchers identified 1,705 ancient viral species based on metagenomic sequencing. About 75% of these viruses were unique to the Giulia Glacier and had never been detected anywhere else on Earth. Only 12% showed similarity to viruses known from other parts of the world, suggesting they might have been transporting to the glacier from distant regions via wind or other atmospheric processes. Less than 1% had been documented in non-glacier environments, highlighting how glaciers and permafrost preserve viral communities that are distinct from those in soils, oceans, or other habitats. The Giulia viruses were predominantly bacteriophages, viruses that infect bacteria rather than eukaryotic cells. This makes sense given the high bacterial abundance in ice and permafrost. Bacteria can remain metabolically active even at sub-freezing temperatures, slowly breaking down organic matter, and where bacteria exist, bacteriophages inevitably follow. The viral communities showed clear differences depending on the climatic period they came from. Viruses from the last glacial stage, when temperatures were coldest, had different genetic signatures than viruses from the Holocene, the current warm period that began approximately 11,500 years ago. The researchers found that viral community composition correlated with climate transitions. When temperatures shifted from cold to warm, viral diversity changed. New viruses appeared, likely introduced by shifting wind patterns that brought microbes from newly accessible regions. Selection pressures changed as well, favoring viruses adapted to warmer conditions or different host populations. The result was a turnover in viral communities, detectable in the ice core record as shifts in genetic composition between adjacent layers. This research provided a window into how viruses respond to climate change over long timescales. The data showed that major climatic events reshaped viral ecosystems, driving evolution and migration. It also demonstrated that viruses are not passive passengers in climate transitions. They actively influence their host organisms, potentially affecting microbial community structure and function, which in turn influences biogeochemical cycles. Another study, examining viral preservation in 700-year-old caribou feces frozen in ice, recovered both DNA and RNA viral genomes. The DNA virus genome was complete and could be cloned and tested for infectivity. Remarkably, when researchers introduced the cloned virus to plant cells, it replicated and spread systemically, proving that genetic information from viruses hundreds of years old could still produce infectious particles. The partial RNA virus genome was more degraded, consistent with the chemical instability of RNA compared to DNA, but enough sequence was recovered to identify the virus as a novel cryptovirus. These findings raised an important point about viral preservation. DNA viruses are far more likely to survive in frozen environments than RNA viruses. DNA is chemically more stable. Its double helix structure provides redundancy, with each strand backing up the other. RNA is single-stranded in most viruses, and the ribose sugar in RNA is more reactive than the deoxyribose sugar in DNA, making RNA more prone to spontaneous degradation. Over thousands of years, RNA viruses in permafrost likely degrade beyond recovery, while DNA viruses can remain intact. This explains why most zombie virus revivals focus on giant viruses, which all have double-stranded DNA genomes. RNA viruses like influenza, coronaviruses, or the agents of Ebola and dengue fever, even if present in permafrost, would not retain infectious capability after millennia. Their genomes would be too fragmented. From a public health perspective, this is reassuring. The most dangerous pandemic viruses in human history, including influenza strains and emerging zoonotic viruses, are RNA-based and unlikely to pose a zombie virus threat from permafrost. However, DNA viruses include many serious human pathogens. Smallpox, caused by Virology, is a DNA virus. It has been eradicated from nature, with the last natural case occurring in 1977, but the virus exists in frozen samples in laboratories and in human remains buried in permafrost. Poxviruses, the family that includes smallpox, are known for their stability. Research has shown that poxvirus DNA remains intact in mummified tissue centuries old. If viable smallpox virus exists in permafrost and that permafrost thaws, there is a theoretical risk of release. The probability of a pandemic from permafrost smallpox is debated. Most virologists consider it very low for several reasons. First, even if the virus is released, it would need to contact a susceptible human population. Permafrost regions are sparsely populated. Second, the virus would need to remain viable during thawing and exposure to environmental conditions that degrade viruses, including UV radiation, oxidation, and microbial degradation. Third, even if someone were infected, modern surveillance and vaccination could contain the outbreak. Smallpox vaccines still exist and can be rapidly produced. The infrastructure to detect and respond to smallpox is robust, having been refined during the eradication campaign. Nevertheless, the discovery of viable 48,500-year-old giant viruses proves that ancient viruses can survive and remain infectious. The prudent course is to monitor thawing permafrost regions for emerging pathogens and to maintain readiness to respond if ancient viruses of public health concern are detected. The 70,000 species estimate specifically refers to distinct viral genotypes that have been detected in permafrost and glacier ice based on metagenomic sequencing. This number will grow as more samples are analyzed. It represents only the viruses whose DNA has been recovered and sequenced. Many viruses in permafrost have likely degraded beyond detection, especially those from the oldest and most damaged ice layers. The true historical diversity of viruses preserved in permafrost may be orders of magnitude higher, with only a fraction recoverable using current technology. The heterogeneity in viral abundance across permafrost samples is puzzling. Why do some samples contain abundant viruses while others have almost none? The answer may relate to the original ecosystem. Samples from lake sediments, where microbial productivity was high and organic matter abundant, tend to have higher viral loads than samples from upland tundra soils. The type of permafrost also matters. Syngenetic permafrost, which forms as sediment accumulates and freezes incrementally, may preserve viruses better than epigenetic permafrost, which forms when existing sediment freezes. The mineral composition, water content, and freezing rate all influence preservation. Another factor is the abundance of the virus's hosts. Giant viruses infect protists like amoebae and algae. If these hosts were abundant in the ancient ecosystem, their viruses would also be abundant. Samples from environments with thriving protist communities would be expected to have high giant virus content. Conversely, environments where protists were scarce would have fewer giant viruses, regardless of how well the ice preserved them. The metagenomic approach has revolutionized our ability to survey viral diversity, but it has limitations. It detects only viruses whose sequences have sufficient similarity to known viral genes to be recognized as viral. Highly divergent viruses, those with genomes completely unlike anything in databases, may be present but undetectable. Researchers call this viral dark matter, sequences that are clearly not bacterial, archaeal, or eukaryotic, but cannot be definitively identified as viral or assigned to any known viral family. A significant fraction of sequences in permafrost metagenomes fall into this dark matter category. They may represent deeply divergent viral lineages, remnants of viral families that went extinct, or entirely novel types of genetic entities. Some researchers speculate that permafrost contains not just known types of viruses, but fundamentally different genetic systems that do not fit into current classifications. If true, the 70,000 species estimate is a dramatic undercount, and the true diversity of viral and virus-like entities in permafrost could be in the millions. The 70,000 hidden species are not static. As permafrost thaws, the viruses within are released into the environment. If their hosts are present, infection can occur, restarting viral life cycles that have been paused for millennia. The thawing Arctic is thus not only releasing greenhouse gases and altering landscapes, it is releasing a hidden biosphere, a frozen seed bank of microbial diversity that is re-entering the modern world. The consequences of this release, both ecological and epidemiological, are only beginning to be explored. Chapter 6: How Ancient Viruses Survive Frozen Time. The fact that a virus can remain infectious after 48,000 years of being frozen challenges our understanding of biological stability. Life is dynamic, constantly renewing and repairing itself. Death is the cessation of those processes, and with it comes decay. Organic molecules break down, proteins denature, DNA degrades. Over centuries and millennia, biological material turns to dust. Yet viruses frozen in permafrost seem to defy this trajectory. They remain intact, their genetic information preserved, their structural proteins functional, ready to infect the moment conditions permit. How is this possible? The answer lies in the unique chemistry of permafrost and the nature of viral particles themselves. Viruses are not alive in the conventional sense. They are packages of genetic information wrapped in a protein shell. They do not metabolize, do not repair damage, do not respond to their environment. Outside a host cell, a virus is essentially a complex organic molecule, a crystal of protein and nucleic acid. And like other molecules, viruses degrade through chemical reactions that require energy, reactants, or catalysts. Permafrost denies viruses all three. Temperature is the most obvious factor. Chemical reaction rates double with every 10 degrees Celsius increase in temperature, a relationship known as the Arrhenius equation. Conversely, cooling slows reactions exponentially. At permafrost temperatures, typically ranging from -5 to -15 degrees Celsius, chemical reactions that would take hours or days at room temperature instead take years or centuries. Molecular motion slows. Enzymes that might degrade nucleic acids are inactive or nearly so. Water, which participates in many degradation reactions, is frozen solid, removing it as a reactive medium. DNA degradation occurs primarily through hydrolysis, the breaking of chemical bonds by reaction with water, and oxidation, the reaction with oxygen or other reactive molecules. Hydrolysis requires liquid water. In permafrost, water is ice, locked into crystalline structures where it cannot react with DNA. Oxidation requires oxygen in a reactive form, often as free radicals produced by UV radiation or metabolic processes. Permafrost is buried, shielded from UV light, and metabolic activity is virtually non-existent at such low temperatures. Oxygen is present, but it is not reactive under these conditions. The result is that DNA molecules remain chemically stable for extraordinarily long periods. Research on ancient DNA preservation has shown that DNA half-life in frozen environments can exceed 500,000 years under ideal conditions, though most estimates are more conservative. A study examining DNA degradation in permafrost mammoth bones found that DNA survives best in regions of dense mineral structure, where it is physically protected from mechanical and chemical stresses. The DNA is fragmented, broken into shorter pieces over time, but recognizable sequences persist. For viral genomes, which are often circular or highly compact, the fragmentation is less of an issue because the entire genome is a single molecule protected within the viral capsid. The viral capsid itself plays a crucial role in preservation. The capsid is a protein shell that encases the viral genome. It is not just a passive container. It is a highly organized structure, often with icosahedral symmetry, that physically shields the DNA from the external environment. The proteins forming the capsid are locked into place by non-covalent interactions, hydrogen bonds, electrostatic attractions, and hydrophobic effects that stabilize the structure. At freezing temperatures, thermal motion that might disrupt these interactions is minimized, and the capsid remains intact, essentially indefinitely. Giant virus capsids are particularly robust. They are large, complex, and often reinforced with lipid membranes and additional protein layers. Mimivirus, for example, has a double-layered capsid with protein fibers extending from its surface and an internal lipid membrane surrounding the genome. This multi-layered protection makes the virus exceptionally resistant to environmental degradation. Pithevirus has a thick-walled capsid with a cork-like structure sealing one end, creating a hermetically sealed environment for the genome. Such structures are well-suited to surviving long-term frozen storage. But protection is not perfect. Even in permafrost, DNA damage accumulates over time. The primary form of damage is spontaneous depurination, the loss of purine bases, adenine and guanine, from the DNA backbone. This reaction occurs even at low temperatures, though very slowly. Each depurination event creates an abasic site, a position in the DNA strand where a base is missing. Abasic sites make DNA prone to breakage. Over thousands of years, depurination can accumulate to the point where the DNA backbone fragments. The rate of depurination depends on temperature and pH. Warmer and more acidic conditions accelerate the reaction. Permafrost pH varies, but many permafrost soils are slightly acidic, which is not ideal for DNA preservation. However, the extreme cold more than compensates. Calculations suggest that at -5°C, depurination rates are roughly 100 times slower than at room temperature. Over 48,000 years, this would result in a manageable level of damage, especially for large genomes where a few abasic sites do not destroy functionality. Another source of DNA damage is ionizing radiation from radioactive isotopes in the surrounding soil. Potassium-40, uranium, thorium, and their decay products emit alpha particles, beta particles, and gamma rays that can damage DNA by creating strand breaks and base modifications. The dose rate depends on the concentration of these isotopes in the permafrost. In typical soils, background radiation causes measurable DNA damage over thousands of years, but the rate is low enough that substantial DNA survives, especially if the genome is protected within a viral capsid. RNA, by contrast, is far less stable. The ribose sugar in RNA has a hydroxyl group that makes the molecule prone to self-cleavage, a reaction called alkaline hydrolysis. Even at neutral pH and low temperature, RNA degrades much faster than DNA. This is why RNA viruses are unlikely to survive in permafrost for tens of thousands of years, while DNA viruses can. The chemical structure of DNA, with its deoxyribose sugar lacking that reactive hydroxyl group, confers long-term stability. Ice crystal formation is another factor influencing preservation. When water freezes, it forms ice crystals that can damage cells and particles by physical disruption. The sharp edges of growing ice crystals can pierce membranes, rupture cells, and fragment large molecules. This is why freezing is often lethal to organisms. However, the damage depends on how fast freezing occurs and how large the ice crystals grow. Rapid freezing produces small ice crystals that cause less mechanical damage. Slow freezing allows large crystals to form, which cause more damage. In permafrost, freezing typically occurs relatively slowly as temperatures gradually drop over seasons or years. This might seem problematic, but in practice, permafrost conditions limit crystal growth. Once soil is frozen, further crystal growth is constrained by the solid matrix. Viruses trapped in frozen soil are embedded in a rigid structure where ice crystals do not continue growing indefinitely. Moreover, viral particles are small and robust. Unlike cells, which have fragile membranes and complex internal structures, viral capsids are solid protein shells. They can withstand the physical stress of ice crystal formation better than cellular organisms. This resilience is part of why viruses survive in permafrost while most cells do not remain viable. There is also the question of whether viruses remain completely dormant or whether some biochemical activity continues at ultra-low temperatures. Research on metabolic activity in permafrost has found that certain bacteria can remain metabolically active, though at incredibly slow rates, even at -20 degrees Celsius. These bacteria can repair DNA damage, slowly synthesizing repair enzymes and maintaining genome integrity over millennia. Could viruses benefit from this if they are inside host cells that are still minimally active? The answer is almost certainly no for viruses outside cells. Free viral particles do not have metabolic machinery and cannot repair themselves. However, viruses integrated into host genomes, like certain bacteriophages and the Mavrus virophage, might benefit from host DNA repair if the host remains active. This would allow integrated viruses to survive even longer than free particles, with their genomes continuously repaired by the host cell's enzymes. In practice, most ancient viruses recovered from permafrost are free particles, not integrated forms. They have survived as inert structures, chemically stable, protected by their capsids frozen in place. When permafrost thaws, whether naturally due to climate change or artificially in a laboratory, the ice melts, the viruses are released, and water becomes liquid again. If a viral particle encounters an appropriate host cell, the infection machinery, dormant for millennia, activates. The capsid disassembles, the genome enters the cell, viral genes are transcribed, and replication begins. The revival experiments conducted by Clavery's team confirmed that this process works even after 48,000 years. The viruses showed no loss of infectivity compared to modern relatives. Infection rates, replication efficiency, and virion production were all normal. The long frozen stasis left no detectable scar on the virus's ability to function. This is a testament to the exquisite molecular stability of viral particles under permafrost conditions. But there are limits. Not all permafrost preserves viruses equally well. Samples from regions that experienced partial thawing and refreezing cycles show greater DNA degradation. Samples from very old permafrost, hundreds of thousands of years or more, have lower recovery rates of intact viral genomes. The DNA is more fragmented, the viral particles more likely to be damaged beyond functionality. The practical limit for recovering infectious viruses from permafrost is likely in the range of 50,000 to 100,000 years, though metagenomic recovery of degraded viral genomes can extend much further. Environmental conditions after thawing also matter. If permafrost thaws and the viruses are exposed to sunlight, UV radiation will rapidly degrade them. If they thaw into water with high microbial activity, enzymes produced by bacteria and fungi can degrade viral nucleic acids. If oxygen is abundant, oxidation reactions will damage proteins and DNA. For a virus to remain infectious after release from permafrost, it must quickly find a host or re-enter conditions that protect it, such as refreezing or encapsulation in sediment. The survival of ancient viruses in permafrost is a combination of chemical stability, physical protection, and environmental luck. The viruses that survive are those with robust capsids, DNA genomes, and the fortune to be frozen in soil that remains undisturbed and deeply cold for millennia. They represent a tiny fraction of the viruses that once existed in ancient ecosystems. Most have degraded beyond recovery. But the ones that remain offer an unparalleled window into viral diversity and evolution across deep time. And as the Arctic continues to warm, these ancient survivors are awakening, released from ice into a world transformed. Chapter 7: Reviving the Dead, Laboratory Resurrection. The process of bringing a 48,000-year-old virus back to life sounds like something from a thriller novel, but the reality is both more methodical and more remarkable. Jean Mitchell-Clavery's laboratory at Aix-Marseille University has perfected the protocols for viral resurrection over more than a decade of work, transforming this extraordinary procedure into a repeatable scientific technique. Each revival experiment follows strict safety procedures, involves multiple verification steps, and ultimately provides a window into the biology of ancient ecosystems frozen in time. The process begins in Siberia, where research teams drill into permafrost at carefully selected sites. These locations are chosen based on geological surveys that identify areas where permafrost has remained continuously frozen for tens of thousands of years. The drilling equipment must be sterile to avoid contaminating samples with modern microorganisms. As the drill penetrates through layers of frozen soil, cores are extracted in sections, each section representing a different time period. The deepest cores, from 30 to 50 meters below the surface, contain the oldest material. These cores are immediately frozen at temperatures below -20 degrees Celsius and shipped to France in specialized containers that maintain freezing conditions throughout transport. Any warming during transit could damage the viral particles or allow modern contamination to occur. Once in the laboratory, the cores are stored at -80 degrees Celsius until processing begins. This ultra-cold storage ensures that ancient viruses remain dormant and that no biological activity occurs that might compromise the sample. When researchers are ready to attempt viral revival, they work in a Biosafety Level 2 laboratory, a controlled environment designed to contain potentially infectious agents. The permafrost sample is thawed under sterile conditions in a biological safety cabinet. Thawing must be controlled and gradual. Rapid temperature changes can damage viral particles through thermal shock and ice crystal reformation. The sample is typically warmed slowly to just above freezing, around 4 degrees Celsius, and maintained at that temperature while processing. The thawed permafrost is a mixture of soil, organic matter, ice crystals, and potentially millions of microbial cells and viral particles. To extract viruses from this complex matrix, researchers add sterile buffer solution and mix thoroughly. The buffer helps separate viral particles from soil particles and dilutes any inhibitory substances that might interfere with infection. The mixture is then centrifuged, spun at high speed to separate components by density. Soil particles and large organic debris settle to the bottom, while viruses remain suspended in the liquid supernatant. This supernatant, containing whatever viruses survived in the permafrost, is the key material for revival experiments. But introducing this crude extract directly to cultured cells would be risky. It might contain unknown pathogens, including bacteria or viruses that could infect humans or other organisms. This is why Clavery's team uses amoebae as the host organism. Amoebae are single-celled protists common in soil and water. Giant viruses that infect amoebae cannot infect human cells, animal cells, or plant cells. They are strictly amoeba-specific. This host restriction provides a crucial safety barrier. The amoeba species used is typically Acanthamoeba casellanii or Acanthamoeba polyphaga, organisms that are easy to culture in the laboratory and serve as hosts for most giant viruses. These amoebae are grown in flasks containing nutrient-rich medium at room temperature. They divide rapidly, creating populations of millions of cells within days. To test whether the permafrost sample contains viable viruses, researchers add small amounts of the viral extract to cultures of healthy amoebae. Then they wait and watch. If viable viruses are present and can infect the amoebae, changes will become visible within hours to days. Under a microscope, infected amoebae begin to show signs of distress. They stop moving normally. Their internal structure becomes disorganized. Within the cytoplasm, viral factories begin to appear, visible as large, distinct compartments where viruses replicate. These factories grow larger as infection progresses, eventually filling much of the cell's interior. After 12 to 72 hours, depending on the virus, the infected amoebae lies, bursting open to release newly formed virus particles. This lysis is dramatic when viewed under the microscope. The cell membrane ruptures, the cell contents spill out, and hundreds or thousands of viral particles are liberated into the medium. These particles can now infect neighboring amoebae, creating a spreading infection through the culture. The appearance of this cytopathic effect, the visible damage to cells caused by viral infection, is the first indication that revival has succeeded. But visual observation is not sufficient to confirm that an ancient virus has been revived. Modern contamination must be ruled out. Perhaps the sample was contaminated during transport with a contemporary giant virus. Perhaps the laboratory environment contained amoeba viruses that accidentally entered the culture. To address these concerns, researchers perform multiple control experiments. They test the permafrost sample before adding it to amoebae to confirm it contains no modern viral contamination. They test the amoeba cultures before infection to ensure they are virus-free. They examine the surrounding laboratory environment for any signs of giant viruses. Most importantly, they characterize the virus that emerges from the infected amoebae using genetic sequencing and compare its genome to all known viruses. If the sequence is novel and distinct from any virus in their laboratory or in public databases, contamination becomes highly unlikely. For Pithavirus cybericum, the 30,000-year-old virus revived in 2014, the genetic sequence confirmed it was a completely new species. Its genome was 610,000 base pairs long and shared only moderate similarity with other known giant viruses. Electron microscopy revealed its distinctive ovoid shape, 1.5 micrometers long, with a thick protein wall and a cork-like structure sealing one end. This morphology was unlike any virus the laboratory had worked with previously. The combination of novel sequence and unique structure provided strong evidence that this was genuinely an ancient virus, not a contaminant. The 2023 study, which revived viruses up to 48,500 years old, employed even more rigorous controls. The team tested 7 different permafrost samples from multiple locations and depths. Each sample was processed independently. If contamination had occurred, it would likely affect only one sample, not all 7. Yet viable viruses were recovered from multiple samples, and each virus had a distinct genetic signature. This reproducibility across independent samples provided powerful evidence that the revivals were authentic. One of the most remarkable aspects of these experiments is that the revived viruses show no loss of fitness. When researchers measure infection efficiency, replication rate, and virion production, ancient viruses perform identically to modern relatives. A 48,000-year-old pithavirus infects amoebae just as effectively as a freshly isolated giant virus would. The burst size, the number of new viral particles produced per infected cell, is normal. The latent period, the time between infection and lysis, is typical. By every measurable criterion, 48,000 years of frozen dormancy left no detectable impact on viral function. This is extraordinary when you consider the molecular detail involved. A functional virus requires that thousands of individual proteins maintain their correct three-dimensional shapes. The capsid proteins must fold properly and assemble into the geometric structure of the viral shell. The DNA replication enzymes must retain their catalytic activity. The transcription machinery must recognize viral genes and produce messenger RNAs. Any significant degradation of these proteins would compromise viral function, yet no such degradation is observed. The implication is that permafrost preservation is nearly perfect at the molecular level. Protein structures remain intact. DNA sequences are undamaged or damaged so minimally that viral repair mechanisms can fix them. The entire complex molecular machine that is a virus particle remains functional across geological timescales. This level of preservation exceeds what most biologists thought possible before these experiments were conducted. The revival experiments also provide opportunities to study viral biology in ways that would be impossible with contemporary viruses. Ancient viruses represent evolutionary snapshots from ecosystems that no longer exist. By comparing ancient viral genomes to modern ones, researchers can directly measure evolutionary chain over known time periods. For example, if a 48,000-year-old virus has a genome that differs from its closest modern relative by a certain number of mutations, you can calculate the mutation rate, changes per base pair per year. Such calculations suggest that giant viruses evolve relatively slowly compared to RNA viruses like influenza or HIV. The mutation rate for DNA viruses is inherently lower because DNA polymerase enzymes, which copy DNA, have proofreading abilities that correct errors. Giant viruses, with their large genomes encoding hundreds of genes, can afford to evolve slowly because they are generalists, capable of infecting multiple host species and adapting to changing environments through their extensive genetic repertoire rather than through rapid mutation. The revival experiments have another important application. They allow researchers to test hypotheses about viral ecology and host range. When a 48,000-year-old virus is revived, it can be tested against different amoeba species to see which ones it can infect. It can be tested against modern giant viruses to see if they compete or interact. It can be exposed to modern virophages to determine if these tiny viral parasites can exploit ancient viral factories. These experiments reveal that ancient viruses behave like their modern counterparts, suggesting that the fundamental interactions between giant viruses, their hosts, and their parasites have remained stable over tens of thousands of years. But the revival experiments also highlight gaps in our knowledge. Not all permafrost samples yield viable viruses. Some samples contain abundant viral DNA, detectable through sequencing, but no infectious particles. Why some viruses remain viable while others do not is unclear. It may relate to the specific preservation conditions in different permafrost layers, the depth at which the sample was frozen, the mineral composition of the soil, or simply chance. Understanding what factors determine viral survival would help predict where the highest-risk ancient pathogens might be preserved. There is also the question of how many different types of viruses can be revived. So far, all successfully revived ancient viruses belong to families that infect amoebae or other protists. This is partly because researchers have focused on these viruses for safety reasons, but it also reflects the fact that protist-infecting giant viruses are abundant in soil and permafrost environments. Viruses that infect bacteria, archaea, or multicellular organisms are also present in permafrost, and some have been detected through metagenomics, but attempts to revive them have been less successful. Bacteriophages, viruses that infect bacteria, should theoretically be revivable using similar methods. Researchers would culture bacteria from permafrost samples, introduce permafrost extracts, and watch for bacterial lysis, indicating phage infection. This approach has been attempted, and some success has been reported, though not with viruses as old as the giant viruses Claverie has revived. The challenge is that many bacteria from ancient permafrost are difficult to culture in the laboratory, and if you cannot culture the host, you cannot revive the virus. Viruses that infect plants or animals pose greater challenges and risks. Reviving a plant virus would require thawing permafrost, extracting the virus, and introducing it to living plant tissue, then observing for infection. The safety concerns escalate because plant viruses can sometimes jump to new plant species, potentially threatening agriculture. Animal viruses are even more problematic. Reviving a virus that might infect mammals, including humans, would require extraordinary biosafety measures and would raise ethical questions about whether such experiments should be conducted at all. For now, the revival experiments remain focused on amoeba-infecting giant viruses, where the combination of scientific value and manageable risk is most favorable. These experiments have demonstrated conclusively that ancient viruses can survive in permafrost for tens of thousands of years and remain infectious. They have provided unprecedented opportunities to study viral biology, evolution, and ecology. And they have issued a clear warning. As permafrost thaws due to climate change, these ancient pathogens will be released into the environment, not in controlled laboratory conditions, but in the wild, where their interactions with modern ecosystems are unpredictable. Chapter 8: Climate Change and the Great Thaw. The Arctic is transforming at a pace that defies historical precedent. Surface air temperatures across the Arctic from October 2024 through September 2025 were the warmest recorded since temperature records began in 1900. The last 10 years represent the 10 warmest years on record for the region. This is not gradual warming. It is accelerating change that is fundamentally altering the physical and biological landscape of the far north. The numbers tell a stark story. The Arctic is warming at more than double the rate of the global average, a phenomenon known as Arctic amplification. While global average temperatures have increased by approximately 1.2 degrees Celsius since pre-industrial times, Arctic temperatures have risen by more than 3 degrees Celsius in many regions, with some areas experiencing increases exceeding 4 degrees. This disproportionate warming results from feedback loops that amplify temperature changes in polar regions. The most significant feedback involves snow and ice albedo. Snow and ice are highly reflective, bouncing up to 90% of incoming sunlight back into space. When snow melts or ice retreats, it exposes darker surfaces, either ocean water or land, which absorb 70 to 90% of sunlight instead of reflecting it. This absorbed energy warms the surface, causing more ice and snow to melt, which exposes more dark surface, creating a self-reinforcing cycle. This albedo feedback has been operating at full force in recent decades, as Arctic sea ice extent has declined by roughly 50% since the 1980s. In March 2025, Arctic winter sea ice reached the lowest annual maximum extent in the 47-year satellite record. The ice that did form was thinner and younger than historical averages. The oldest, thickest Arctic sea ice, more than 4 years old, has declined by over 95% since the 1980s. Multi-year ice, which once dominated the Arctic Ocean, is now confined to a small region north of Greenland and the Canadian archipelago. The loss of this thick ice makes the remaining ice more vulnerable to melting during summer, as thin ice breaks apart and melts more easily than thick ice. But the transformation extends beyond sea ice to the frozen ground itself, permafrost. Ground that remains frozen year-round for at least 2 consecutive years underlies approximately 1/5 of the Northern Hemisphere's land surface. This includes vast areas of Siberia, Alaska, northern Canada, Greenland, and parts of Scandinavia. Permafrost can extend hundreds of meters deep, representing millions of years of frozen accumulation. In some regions of Siberia, permafrost reaches depths exceeding 600 meters. The active layer, the uppermost zone that thaws each summer and refreezes each winter, is growing thicker. Historical measurements show active layer depths of 30 to 50 centimeters in many Arctic regions. Recent measurements show depths of 60 to 100 centimeters or more, representing a doubling of the thaw depth in some locations. This deeper seasonal thaw brings previously frozen soil into the active zone, exposing organic matter that has been frozen for millennia to microbial decomposition. More concerning is the thaw of permafrost that has been continuously frozen since the Pleistocene. A study published in 2025 estimated that the permafrost area in the Panarctic declined from 13.4 million square kilometers in the period 2003 to 2013 to 12.51 million square kilometers in 2014 to 2023. This represents a loss of approximately 890,000 square kilometers of permafrost in just 1 decade, an area larger than the entire state of Texas. The thaw is not uniform. Some regions are experiencing rapid, catastrophic collapse, while others show more gradual warming. In Siberia, particularly the Yamal Peninsula and Yukachi-Alas region, massive thermokarst features are forming. Thermokarst occurs when ice-rich permafrost melts, causing the ground to collapse and create depressions, lakes, and craters. Some of these craters are enormous, measuring hundreds of meters across and tens of meters deep. When permafrost containing large ice wedges melts, the resulting subsidence can be dramatic, with the ground dropping several meters in a single season. These landscape changes are visible from space. Satellite imagery shows the proliferation of thermokarst lakes across Siberia and Alaska, appearing as dark blue patches in formerly solid tundra. Some lakes are expanding, merging with neighbors to create larger water bodies. Others are draining as underlying permafrost thaws and create subsurface channels that allow water to escape. The result is a patchwork landscape in constant flux, with wetlands expanding and contracting, rivers changing course, and ground stability becoming unpredictable. The thaw releases enormous quantities of carbon. Permafrost soils contain an estimated 1,600 gigatons of organic carbon, nearly twice the amount currently in Earth's atmosphere. This carbon is locked in plant material, animal remains, and microbial biomass that accumulated over tens of thousands of years. When permafrost thaws, bacteria and fungi colonize the newly available organic matter and begin breaking it down through respiration, releasing carbon dioxide, and through fermentation in waterlogged conditions, releasing methane. Methane is particularly concerning because it is a potent greenhouse gas with a global warming potential 25 times greater than carbon dioxide over a 100-year timescale. When permafrost thaws into waterlogged conditions, creating anoxic environments where oxygen is scarce, methane-producing microbes thrive. Studies measuring methane emissions from thawing permafrost regions show that emissions are increasing, with some sites showing methane flux densities 10 to 100 times higher than historical baselines. The carbon release creates a positive feedback loop. Thawing permafrost releases carbon dioxide and methane, which enhance global warming, which causes more permafrost to thaw, which releases more carbon. This feedback is not yet fully incorporated into climate models, meaning current projections of future warming may underestimate the actual temperature increases we will experience. Some estimates suggest permafrost carbon feedback could add an additional 0.2 to 0.5 degrees Celsius of warming by 2100, potentially pushing global temperatures above critical thresholds. The infrastructure impacts are already severe. Nearly 70% of Russia's energy infrastructure in Arctic regions is built on permafrost. This includes oil fields, gas pipelines, storage facilities, and transportation networks. As permafrost thaws, the ground becomes unstable, pipelines buckle and rupture, storage tanks tilt and crack, buildings collapse, roads become impassable as the ground beneath them turns to mud or develops sinkholes. In 2025, a study estimated that building and road losses due to permafrost thaw in Alaska alone could cost between $37 billion and $51 billion by mid-century under moderate and high emissions scenarios. Similar costs are projected for Canada and Russia, where permafrost infrastructure is even more extensive. The economic impact extends beyond direct damage to include increased maintenance costs, more frequent repairs, shortened infrastructure lifespan, and the need for expensive engineering solutions like thermosiphons and elevated building foundations. Communities built on permafrost face existential threats. Villages in Alaska, Siberia, and northern Canada are experiencing catastrophic ground instability. Homes sink as permafrost beneath them melts. Water and sewage systems rupture. Coastal erosion accelerates as permafrost cliffs thaw and collapse into the ocean. Some communities are being forced to relocate entirely, becoming climate refugees. The village of Shishmeef in Alaska has voted to relocate because erosion and permafrost thaw have made the current location uninhabitable. Similar relocations are planned or underway in dozens of Arctic communities. The pace of change is accelerating. Autumn 2024 and winter 2025 were the first and second warmest on record for the Arctic. Snow cover, which had above-average extent during winter 2025, melted so rapidly in spring that by June, coverage was significantly below historical averages. This swift transition from heavy snow to bare ground is characteristic of a climate system undergoing rapid change, where extreme variability replaces stable seasonal patterns. The Greenland ice sheet is losing mass at an accelerating rate, contributing to global sea level rise. Mountain glaciers across the Arctic are retreating 3 times faster than in the 1990s. The Mendenhall Glacier in Alaska has retreated so far that glacial lake outburst floods, sudden catastrophic releases of meltwater, have inundated homes in Juneau multiple times in recent years. These floods occur when ice dams holding back glacial lakes fail as the ice thins and weakens. Rivers draining Arctic watersheds are changing color, turning orange and rust-colored due to increased metal mobilization from thawing permafrost. As permafrost thaws, minerals that have been locked in frozen soil for millennia are exposed to oxygen and water, triggering chemical reactions that release iron, zinc, copper, and other metals. These metals enter streams and rivers, altering water chemistry and potentially contaminating drinking water sources for Arctic communities. The biological impacts are equally profound. As permafrost thaws, the frozen archive of ancient microorganisms is released. Bacteria that have been dormant for tens of thousands of years become active again when exposed to liquid water and warmer temperatures. Some of these bacteria are harmless, others are pathogens. The 2016 anthrax outbreak in Siberia demonstrated that ancient bacterial pathogens can survive in permafrost and cause disease when released. That outbreak occurred in the Yamal Peninsula during an exceptionally hot summer. Temperatures exceeded 35 degrees Celsius, far above normal for the region. The heat wave thawed permafrost that had been frozen for over 70 years, exposing a reindeer carcass infected with anthrax. The carcass had been buried in shallow permafrost during an anthrax outbreak in the 1940s. When it thawed, viable anthrax spores were released into the environment. Reindeer grazing near the thaw site ingested the spores. Within days, animals began dying. The outbreak spread rapidly through herds, eventually killing more than 2,000 reindeer. Humans handling the sick animals or their carcasses became infected. 72 people were hospitalized, and a 12-year-old boy died. The outbreak was contained only through massive veterinary and public health response, including quarantine, vaccination, and carcass disposal. This event was a wake-up call. If anthrax spores can survive 70 years in permafrost and remain virulent, what else might be preserved? Historical records document numerous anthrax outbreaks in Siberia during the 19th and early 20th centuries. Burial sites from those outbreaks contain infected carcasses, and those sites are now thawing. Russia has identified approximately 7,000 such burial grounds across northern regions. Each represents a potential source of future anthrax release. But anthrax is just one pathogen. Smallpox, which was endemic in Siberia before its eradication, could theoretically be preserved in human remains buried in permafrost. Plague, which caused major outbreaks in Siberia during the 19th century, might also be present. While the likelihood of these viruses remaining viable is debated, the possibility cannot be dismissed. The longer permafrost has been frozen, the more likely it contains ancient pathogens that modern populations have never encountered. The thaw also releases ancient viruses that infect soil microorganisms. As discussed in previous chapters, giant viruses and bacteriophages are abundant in permafrost. When released, these viruses encounter modern microbial communities. If they can infect modern hosts, they may influence ecosystem processes like nutrient cycling and decomposition. If they cannot, they simply become inactive again. The ecological consequences of releasing tens of thousands of ancient viral species into modern environments are unknown and virtually impossible to predict. The window for studying pristine permafrost is closing. Every year, more permafrost thaws, releasing its contents. Every summer, deeper layers are exposed to warmth and microbial activity. The frozen archive that has preserved a record of ancient life for tens of thousands of years is degrading. Within decades, large regions of permafrost that have been continuously frozen since the Pleistocene will have thawed completely. The opportunity to study this archive will be lost, and with it, insights into how ecosystems functioned during ice ages, how microbial communities evolved over deep time, and what diversity of life existed in ancient Arctic environments. We are witnessing the Great Thaw, a transformation of the Arctic that will reshape the planet. The impacts extend far beyond the polar regions. Carbon released from permafrost will accelerate global warming, affecting climate worldwide. Methane emissions will intensify. Sea levels will rise as ice sheets melt. Weather patterns will shift as the temperature gradient between the Arctic and lower latitudes changes. And ancient viruses, bacteria, and other microorganisms will emerge from their frozen tombs, entering a world utterly transformed from the one they last inhabited 48,000 years ago. Chapter 9: The Pandemic Risk, Fact versus Fiction. The image of ancient viruses emerging from melting ice to cause a global pandemic is compelling and terrifying, but how realistic is this scenario? Media coverage of zombie viruses often emphasizes catastrophic possibilities, with headlines warning of prehistoric plagues and unstoppable outbreaks. The scientific reality is more nuanced and considerably less apocalyptic, though genuine risks do exist that warrant careful monitoring and preparedness. First, it is essential to understand what types of viruses are actually being revived from permafrost. All viruses that Jean Mitchell-Clavery's team has successfully revived and confirmed as infectious are giant viruses that specifically infect amoebae. These viruses cannot infect human cells, animal cells, or plant cells. They are strictly limited to protist hosts, primarily free-living amoebae like Acanthamoeba species. The host specificity of viruses is one of their defining characteristics. A virus evolves to recognize and bind to specific receptor molecules on its host cell's surface. Without those receptors, the virus cannot attach, cannot enter, and cannot infect. Amoeba-infecting giant viruses use receptors that are unique to protist cell surfaces. Human cells lack these receptors entirely. Even if a person was somehow exposed to billions of pithevirus particles, perhaps by drinking water containing thawed permafrost, the viruses would pass through the body without causing infection. They would be unable to recognize human cells, unable to attach, unable to penetrate cell membranes. This fundamental biological barrier provides substantial reassurance that the viruses currently being revived pose no direct pandemic threat. However, permafrost does not exclusively contain amoeba viruses. Metagenomic studies reveal diverse viral communities, including bacteriophages that infect bacteria, viruses that infect algae and other protists, and potentially viruses that once infected animals. The question becomes whether any of these other viruses could be revived and pose risks to modern organisms, including humans. Consider bacteriophages, which are abundant in permafrost. These viruses infect bacteria and are generally harmless to humans. In fact, bacteriophages are so specific that a phage that infects one bacterial species typically cannot infect even closely related species. The idea of a bacteriophage causing human disease is not scientifically plausible. Bacteriophages have been used therapeutically to treat bacterial infections in humans for nearly a century, with no adverse effects from the phages themselves. Viruses that infect algae and other photosynthetic organisms are similarly specific and pose no threat to humans. The same applies to viruses infecting fungi, archaea, and most other microorganisms. The overwhelming majority of viruses in permafrost, even if revived, would be ecologically interesting but medically irrelevant. The concern centers on viruses that might have infected mammals, including humans or their ancestors, tens of thousands of years ago. These viruses would theoretically possess the molecular machinery to recognize mammalian cells and initiate infection. If such viruses remain viable in permafrost and are released as the Arctic thaws, could they cause outbreaks? Several factors make this scenario unlikely, though not impossible. First, viruses that infect mammals are mostly RNA viruses, including influenza, coronaviruses, measles, mumps, and most emerging zoonotic viruses. As discussed in earlier chapters, RNA is chemically unstable compared to DNA. RNA viruses frozen in permafrost for thousands of years would experience extensive degradation of their genomes, rendering them non-infectious. The spontaneous cleavage of RNA under freezing conditions, though slow, accumulates over millennia to the point where genomic integrity is lost. This has been confirmed experimentally. Studies attempting to recover RNA virus genomes from ancient frozen samples find only fragments, short sequences that can be identified through sequencing but cannot produce infectious virus. A study of 700-year-old caribou feces frozen in ice recovered partial RNA virus genomes, but the sequences were incomplete and could not be reconstructed into viable viruses. 48,000 years is vastly longer than 700 years, making RNA virus survival even more improbable. DNA viruses, however, are more stable. Several families of DNA viruses infect mammals, including poxviruses like smallpox, herpes viruses, adenoviruses, and papillomaviruses. Of these, poxviruses are the most concerning from a pandemic perspective because they include variola virus, which caused smallpox and killed hundreds of millions of people throughout history. Smallpox was declared eradicated in 1980 after a global vaccination campaign eliminated the virus from nature. The last natural case occurred in Somalia in 1977. Official stocks of variola virus are maintained in 2 high-security laboratories, one in the United States and one in Russia. But unofficial stocks may exist in forgotten freezers, and the virus may be preserved in human remains buried in permafrost regions where smallpox was endemic. Historical records document smallpox outbreaks in Siberia during the 18th and 19th centuries. Bodies of smallpox victims were buried, often in shallow grave, in permafrost soils. If those bodies remain frozen, the virus could theoretically be preserved. Poxviruses are known for their stability. Research in the 1950s showed that vaccinia virus, used in smallpox vaccines, survives freezing for years. Whether variola virus could survive tens of thousands of years is unknown, but it cannot be ruled out entirely. In 1997, researchers exhumed bodies from a 19th-century graveyard in Alaska where smallpox victims had been buried in permafrost. The goal was to recover viral DNA for historical study, not to revive the virus. The bodies were remarkably preserved after more than a century, but when tissue samples were tested, no viable smallpox virus was detected. Viral DNA was present, but it was fragmented and degraded. The researchers concluded that even in permafrost, the virus had not remained infectious over a century. This finding is reassuring, but not definitive. A century is far shorter than the timescales relevant for ancient permafrost. Moreover, preservation conditions vary. Deeper, colder permafrost that has never experienced seasonal thaw might preserve viruses better than shallow permafrost near the surface. The mineral composition of soil, the presence of protective organic matter, and the initial viral load in the buried individual all influence preservation. Even if viable smallpox virus were released from thawing permafrost, several barriers would prevent it from causing a pandemic. First, the virus would need to reach a susceptible human host. Permafrost regions are sparsely populated. The probability of thawed virus particles directly contacting a person is low. Second, smallpox transmission requires close contact with an infected individual or their bodily fluids. A few virus particles released into soil would likely be inactivated by environmental factors like UV radiation, desiccation, and microbial degradation before reaching a host. Third, even if a person were infected, modern surveillance systems would likely detect the outbreak quickly. Smallpox has a distinctive clinical presentation with characteristic lesions that are easily recognized. Health authorities worldwide remain vigilant for any smallpox case because of bioterrorism concerns. An isolated case would trigger immediate response, including isolation, contact tracing, and vaccination of exposed individuals. Smallpox vaccines still exist and can be manufactured rapidly if needed. Fourth, vaccination has left some residual immunity in populations. People vaccinated before 1980, when routine smallpox vaccination ended, may retain partial immunity decades later. While this immunity might not prevent infection entirely, it could reduce disease severity and transmission. Younger individuals have no immunity, which is concerning, but the infrastructure to respond exists. The 2016 anthrax outbreak in Siberia provides a real-world case study of pathogen release from thawing permafrost. Anthrax is a bacterial disease caused by Bacillus anthracis, which forms extremely durable spores that can survive centuries in soil. When thawed, the spores caused an outbreak affecting thousands of reindeer and dozens of people. However, the outbreak was contained through veterinary intervention, quarantine, and human treatment with antibiotics. One death occurred, tragic but far from a pandemic. Anthrax is arguably more dangerous than most ancient viruses because the spores are environmentally stable and can remain infectious for decades after release, whereas viruses typically degrade quickly once exposed to sunlight, oxygen, and fluctuating temperatures. If anthrax can be contained, zombie viruses likely can as well. The greater uncertainty lies not with known pathogens like smallpox or anthrax, but with unknown pathogens. Permafrost contains viral and bacterial species that have never been characterized. Metagenomic surveys reveal vast viral dark matter, sequences that cannot be matched to any known organism. Some of these unknown viruses might have broad host ranges or unusual transmission mechanisms. Assessing risk for something completely unknown is inherently difficult. Expert opinion on pandemic risk from permafrost viruses is generally cautious but not alarmist. Virologists emphasize that while the theoretical risk exists, the practical probability remains low. The majority of ancient viruses cannot infect humans. Those that can would face multiple barriers to causing widespread disease. However, as the Arctic warms and human activity in polar regions increases, exposure opportunities grow. Mining operations, oil and gas extraction, infrastructure development, and tourism all bring people into contact with thawing permafrost. One scenario of concern involves laboratory workers. Scientists studying permafrost samples could theoretically be exposed to viable pathogens during sample processing. This is why biosafety protocols are essential. Clavery's laboratory adheres to strict procedures, including working in biosafety cabinets, using personal protective equipment, and limiting experiments to amoeba-infecting viruses. For any work involving potential human or animal pathogens, biosafety level 3 or 4 facilities would be required, with additional layers of containment and safety measures. Another scenario involves accidental environmental release. If permafrost thaws naturally, any pathogens it contains are released into the environment without human intervention. Animals grazing on newly thawed ground, drinking from meltwater ponds, or scavenging thawed carcasses could become infected. If the pathogen can infect domestic animals or wildlife that interact with humans, a transmission chain could begin. This is how the 2016 anthrax outbreak started. Reindeer grazed near a thawed carcass, became infected, and then transmitted the disease to humans handling the sick animals. Monitoring is the key to managing this risk. Public health agencies in Arctic nations are beginning to establish surveillance systems to detect emerging pathogens in permafrost regions. This includes monitoring animal health, testing water sources, and conducting environmental sampling for pathogens. Early detection allows rapid response before an outbreak becomes widespread. Climate change itself complicates risk assessment. A warmer Arctic means more human activity, longer summer seasons when thaw is extensive, and potentially more permafrost disturbance through construction and resource extraction. It also means that environments previously too cold for year-round human habitation may become accessible, bringing more people into contact with thawing ground. The pandemic risk from ancient viruses exists on a spectrum. At one end, amoeba-infecting giant viruses pose essentially zero risk to human health. At the other end, viable human pathogens like smallpox, if they exist in permafrost and are released, could cause localized outbreaks requiring significant public health response, but likely would not cause pandemics given modern surveillance and response capabilities. In between lies uncertainty about unknown pathogens and novel viruses whose behavior we cannot predict. The scientific consensus is that monitoring and preparedness are warranted, but panic is not. The zombie virus narrative makes for dramatic headlines, but the reality is more measured. Ancient viruses emerging from permafrost are a concern worth studying and watching, but they are not an imminent apocalyptic threat. The greater climate-related health risks remain the established impacts of warming, heat waves, changing disease vector distributions, food and water security, and the social disruption caused by climate migration and infrastructure collapse. Zombie viruses are part of a broader climate crisis, not a separate catastrophe. Chapter 10: Viral Dark Matter and Unknown Diversity. When scientists sequence DNA from environmental samples like ocean water, soil, or permafrost, they make a startling discovery. The majority of sequences do not match anything in existing databases. These mysterious genetic sequences represent organisms, genes, and biological functions that science has never encountered before. Nowhere is this phenomenon more pronounced than in the viral world, where as much as 95% of sequences detected in metagenomic studies have no recognizable similarity to known viruses. Scientists call this the viral dark matter, an apt name that evokes the mysterious dark matter of cosmology that makes up most of the universe, but remains invisible and poorly understood. The concept emerged in the early 2000s as high-throughput DNA sequencing technology became affordable and widespread. Previously, identifying viruses required culturing them in the laboratory, growing them in host cells, isolating viral particles, and characterizing them one at a time. This approach worked for viruses that could be cultured, but the vast majority of viruses cannot be grown in laboratory conditions. Their hosts are unknown, or their hosts cannot be cultured, or the precise environmental conditions needed for infection cannot be replicated. These unculturable viruses remained invisible to science. Metagenomics changed everything. Instead of culturing organisms, researchers could simply extract all DNA from an environmental sample, sequence it, and use computational tools to identify which sequences belong to viruses based on characteristic features like genome organization, gene content, and sequence similarity to known viral genes. This culture-independent approach opened a window into microbial diversity that had been completely hidden, but it also revealed how little we actually know about the viral world. Consider a typical metagenomic study of ocean water. Researchers filter seawater to concentrate viral particles, extract their DNA, sequence it, and analyze the resulting data. The sequences are compared against GenBank, the largest public repository of genetic information, which contains hundreds of billions of base pairs from millions of organisms. In a typical viral metagenome from seawater, between 60 and 95% of sequences have no match in GenBank. They are orphans, genetic information without context, origin, or function. Some of these orphan sequences can be identified as viral through indirect evidence. They might be found within genomic scaffolds that contain a few recognizable viral genes, like those encoding major capsid proteins or DNA polymerases. The presence of these hallmark genes suggests the entire scaffold is viral, even if most genes on it are unknown. Other sequences lack even these clues. They are simply stretches of DNA encoding proteins that have never been seen before, with no obvious relationship to anything known to science. This viral dark matter poses fundamental questions. How much viral diversity actually exists on Earth? Current estimates suggest that tens of millions to hundreds of millions of distinct viral species exist globally. A conservative back-of-the-envelope calculation starts with the number of cellular species. There are approximately 1 trillion species of bacteria and archaea on Earth based on recent estimates. If each bacterial species is infected by an average of 10 distinct virus types, that yields 10 trillion viral species. Even accounting for the fact that some viruses infect multiple host species, reducing this number, the total viral diversity would still be measured in the billions. But these estimates are highly uncertain because they depend on assumptions about host range, viral specificity, and the relationship between host diversity and viral diversity. The true number could be higher or lower by orders of magnitude. What is certain is that known viral diversity represents a tiny fraction of total diversity. As of 2026, the International Committee on Taxonomy of Viruses recognizes approximately 11,000 virus species. This is the number that have been isolated, cultured, characterized, and formally described. Metagenomic studies suggest this represents less than 1% of existing viral diversity. The 70,000 species estimate for permafrost and glacial ice is based on metagenomic data from multiple studies conducted over the past decade. Researchers analyzing permafrost samples have identified tens of thousands of distinct viral genotypes, genetic variants that differ from each other by more than 5 to 10% at the nucleotide level, a threshold commonly used to delineate species boundaries in viruses. These genotypes are detected based on assembly of metagenomic reads into scaffolds and comparison of those scaffolds against databases of known viral sequences. In the 2022 study of Siberian permafrost, 721 distinct scaffolds belonging to giant viruses were identified from just 11 samples. Each scaffold potentially represents a different species or strain. Extrapolating from these 11 samples to the millions of square kilometers of permafrost across the Arctic, the total diversity could easily reach hundreds of thousands or millions of distinct giant virus species alone. Adding bacteriophages and other viral types increases the estimate further. The Tibetan glacier study identified 1,705 viral species from ice cores spanning 15,000 years, with 75% of them unique to that single glacier. If each glacier worldwide contains a similarly unique viral community, the diversity locked in ice is immense. And these are just the viruses whose DNA sequences are sufficiently intact and abundant to be detected through sequencing. Degraded, rare, or highly divergent viruses would be missed, meaning the true diversity is even higher. The concept of viral dark matter extends beyond simple detection. Even when a viral sequence is identified as viral, understanding what it does is another challenge entirely. Viral genomes are replete with genes encoding proteins of unknown function. In Pandaravirus, 93% of genes have no similarity to anything in databases, meaning we have no idea what these genes do, what proteins they encode, or what role they play in the viral life cycle. The virus was discovered, characterized structurally, and shown to infect amoebae, but its biology remains largely mysterious. This is not unique to Pandaravirus. Across all giant viruses, roughly half to three-quarters of genes are orphans, encoding hypothetical proteins with no known function. Some of these genes may be involved in metabolism, manipulating host cells, evading immune responses, or regulating gene expression. Others might perform functions we have never imagined because they represent strategies unique to viruses, strategies that cellular organisms do not use, and that therefore have no precedent in our databases built primarily from cellular life. The challenge of viral dark matter has spurred development of new computational tools designed to identify viral sequences even when they have no similarity to known viruses. One approach looks for genome organization, patterns characteristic of viruses. Viruses tend to have compact genomes with little non-coding DNA, genes densely packed together, and specific arrangements of genes related to DNA replication, capsid structure, and genome packaging. Machine learning algorithms can be trained to recognize these patterns and flag sequences as likely viral even if the individual genes are unknown. Another approach uses protein structure prediction. Even if a protein sequence has no similarity to known proteins, its three-dimensional structure might resemble known structures. Recent advances in artificial intelligence, particularly tools like AlphaFold, allow researchers to predict protein structures from amino acid sequences with remarkable accuracy. By predicting structures for proteins encoded by dark matter genes, Scientists can sometimes identify functional domains or structural motifs that provide clues about what the protein does. A third approach looks for contextual clues. If an unknown gene is consistently found adjacent to known viral genes across multiple genomes, it is likely viral and probably functionally related to those neighboring genes. Guilt by association is not definitive proof, but it helps narrow possibilities and generate hypotheses that can be tested experimentally. Despite these tools, progress in characterizing viral dark matter is slow. The sheer volume of unknown sequences is overwhelming. Every new metagenomic study adds thousands to millions of new sequences, most of them orphans. Databases grow faster than our ability to annotate and characterize them. We are in an era of biological discovery where data generation vastly outpaces data interpretation. This situation is both exciting and frustrating. Exciting because the viral world is far richer and more diverse than anyone imagined 2 decades ago. Frustrating because we lack the resources and methods to fully explore this diversity. Each permafrost sample contains genetic information from viruses that may have ecological functions we cannot predict, evolutionary histories we cannot trace, and biological capabilities we cannot fathom. Some scientists have proposed that viral dark matter includes entirely novel types of genetic entities that do not fit into current classifications. Perhaps there are viruses with fundamentally different genome organizations, replication strategies, or capsid structures that are so alien to known viruses that we cannot recognize them as viruses at all. They would appear in metagenomic data as anomalous sequences, present but unclassifiable. Others suggest that much of the dark matter represents rapidly evolving genes under strong selection pressure. Viruses engaged in arms races with host immune systems evolve quickly, with genes involved in immune evasion changing so fast that they share no detectable similarity across even closely related viruses. These genes would appear as orphans in every genome, each virus having evolved its own unique set of immune evasion strategies. Still others propose that dark matter includes genes borrowed from extinct organisms. If a virus infected a host species that is now extinct and incorporated genes from that host into its genome, those genes would have no match in modern databases built from living organisms. The virus becomes a genetic fossil, carrying remnants of vanished biodiversity. The 70,000 viral species in permafrost represent a mixture of all these categories. Some are relatives of known viruses, divergent but recognizably related. Others are deeply divergent, belonging to families not yet discovered in modern environments. And some may be representatives of viral lineages that went extinct everywhere else, but persist in the frozen archive. The unknown sequences in permafrost viromes also raise questions about functional diversity. Viruses are not just parasites, they are ecological engineers that influence nutrient cycling, population dynamics, and ecosystem structure. Giant viruses, for instance, alter host metabolism during infection, reprogramming cells to produce viral components. If the genes involved in this metabolic manipulation are unknown, we cannot predict how viral infection influences ecosystem processes. Thawing permafrost could release viruses that interact with modern microbial communities in ways we cannot anticipate because we do not understand their biology. Characterizing viral dark matter will require decades of research and significant investment in both experimental virology and computational biology. Laboratory experiments are needed to culture novel viruses, test gene functions, and validate computational predictions. Computational tools must be refined to better identify viral sequences and predict functions from minimal information. International collaboration is essential because viral diversity spans the globe, and no single laboratory or nation can catalog it alone. The viral dark matter is not just an academic curiosity, it represents the largest reservoir of genetic innovation on the planet. Viruses have been exchanging genes with cellular organisms for billions of years, and many of those genes now reside in bacterial, archaeal, and eukaryotic genomes. Understanding viral dark matter means understanding the origins of a substantial fraction of cellular genetic diversity, including genes involved in metabolism, immunity, and development. In permafrost, the dark matter is frozen alongside the known. Every sample contains sequences that match giant viruses in databases and sequences that match nothing. As the Arctic thaws, both are released. The known viruses can be studied, characterized, and monitored. The unknown viruses remain enigmatic, genetic ghosts emerging from ice into a world that has no context for them. They are the 70,000 species hiding in plain sight, visible as genetic sequences, but invisible in terms of biology, ecology, and evolution. They are the frontier of virology, the next chapter in a story we have only just begun to read. Chapter 11: Evolution written in frozen genomes. Permafrost is not just a freezer preserving ancient viruses. It is a time machine that allows scientists to directly observe evolution across tens of thousands of years. By comparing viral genomes from different time periods within permafrost cores, researchers can trace how viruses have changed, which genes have remained stable, which have evolved rapidly, and how viral communities have responded to environmental shifts. This paleogenomics approach provides insights into evolutionary processes that would be impossible to obtain through any other method. The basic principle is straightforward. A permafrost core represents a vertical timeline. The surface layer contains modern viruses from organisms living today or recently. Deeper layers contain viruses from progressively older time periods. By dating each layer using radiocarbon dating or other geochronological methods, researchers can assign ages to the viruses found within. If the same viral lineage is found at multiple depths, representing different ages, direct comparisons reveal evolutionary changes over known timescales. The 2022 study that assembled a complete 1.6 million base pair Pithaviridae genome from 42,000-year-old permafrost provided the first opportunity for such analysis. The researchers compared this ancient genome to modern Pithavirus cybricum, which was revived from 30,000-year-old permafrost and represents a closely related but distinct lineage. Despite being separated by at least 12,000 years, the genomes showed remarkable conservation in their core genes, those encoding DNA polymerase, major capsid proteins, and other essential functions. The conservation suggests these core genes are under strong purifying selection, meaning natural selection eliminates mutations that alter their function. This makes sense because these genes are critical for viral survival. A mutation that damages DNA polymerase function would prevent the virus from replicating its genome, eliminating that mutant from the population. Only mutations that maintain or improve function persist. The result is that core genes evolve slowly, changing only when mutations provide some advantage without compromising essential functions. However, other regions of the genomes showed significant divergence. Genes located near the genome ends, often encoding hypothetical proteins of unknown function, differed substantially between the ancient and modern viruses. Some genes present in one genome were absent in the other. Gene order was rearranged in certain regions. These differences suggest that accessory genes, those not essential for basic replication, but potentially involved in host range, immune evasion, or environmental adaptation, evolve much more rapidly than core genes. This pattern of conserved cores and variable peripheries is common in viral evolution and reflects different selective pressures. Core genes must remain functional under all conditions, so they are constrained. Accessory genes can be gained, lost, or modified without destroying the virus's ability to replicate, so they are free to evolve rapidly in response to changing environments or host defenses. Phylogenetic analysis places the ancient Pithaviridae genome within a clade that includes modern Pithavirus species, but as a distinct lineage that diverged before the modern species split from each other. This suggests the ancient virus represents a snapshot of Pithaviridae diversity as it existed 42,000 years ago, before subsequent evolution and environmental changes winnowed diversity down to the lineages we see today. It is possible that dozens or hundreds of Pithaviridae species existed during the Pleistocene, most of which went extinct as ecosystems changed, leaving only a few lineages to survive into the present. Gyulya Glacier study from the Tibetan Plateau provides another perspective on viral evolution through time. The ice cores spanned 15,000 years and captured three major climate transitions from glacial to interglacial conditions. Viral community composition changed across these transitions, with certain viral families becoming more abundant during warm periods and others during cold periods. These shifts reflect changes in host communities, which in turn were driven by climate. During glacial periods, when temperatures were colder and ice coverage more extensive, bacterial communities in the glacier were dominated by psychrophiles, organisms adapted to cold conditions. The viruses infecting these bacteria, primarily bacteriophages, were correspondingly abundant. When temperatures warmed during interglacial periods, different bacterial species colonized the glacier environment, and their associated bacteriophages replaced the cold-adapted viruses. The result was community turnover detectable in the viral metagenomic data. But some viral lineages persisted across all climate transitions. These generalist viruses apparently had broad enough host ranges or sufficiently adaptable strategies to survive in both cold and warm conditions. Phylogenetic analysis of these persistent lineages shows minimal genetic change over the 15,000-year span, suggesting they occupy stable ecological niches that have remained available throughout climate fluctuations. Other viral lineages show evidence of rapid evolution. Genes involved in recognizing and binding to host cells, often located in regions encoding capsid proteins or tail fibers in bacteriophages, accumulate mutations at rates far exceeding the genome-wide average. This rapid evolution is driven by the need to track host evolution. As bacteria evolve resistance to viral infection by modifying surface receptors, viruses must counter-evolve to recognize the modified receptors. This coevolutionary arms race leaves a molecular signature, elevated mutation rates in genes mediating virus-host interactions. Horizontal gene transfer is another evolutionary mechanism visible in permafrost viral genomes. Viruses can acquire genes from hosts or from other viruses, incorporating new genetic material into their genomes. Evidence for horizontal transfer comes from phylogenetic incongruence, where a gene tree conflicts with the species tree. For example, if a gene in a pithavirus genome is more closely related to a eukaryotic gene than to genes in other viruses, horizontal transfer from a eukaryotic host to the virus is the likely explanation. The Permafrost metagenomic study found extensive evidence of horizontal gene transfer between viruses and cellular organisms. Aminosyl tRNA synthetase genes in giant viruses, for instance, show phylogenetic relationships indicating they were acquired from eukaryotes multiple independent times. Different giant virus families acquired different sets of synthetases from different eukaryotic lineages, suggesting ongoing gene capture across millions of years of evolution. Horizontal transfer between different viral families is also detectable. Genes encoding DNA repair enzymes, transcription factors, and metabolic enzymes appear in viruses from distantly related families with nearly identical sequences, indicating recent transfer. The mechanism likely involves co-infection, where two different viruses infect the same cell simultaneously. During replication, genomic recombination can occur, shuffling genes between the two viral genomes and creating hybrid progeny. This genetic mosaicism makes reconstructing viral evolutionary history challenging. Unlike cellular organisms, which primarily inherit genes vertically from parent to offspring, viruses actively exchange genes horizontally, creating patchwork genomes with genes from diverse sources. A single giant virus genome may contain genes inherited from an ancient viral ancestor, genes acquired from eukaryotic hosts within the past million years, genes stolen from bacteria, and genes transferred from other viruses during recent co-infections. Despite this complexity, certain patterns emerge. core genes involved in DNA replication and virion structure tend to show vertical inheritance, allowing construction of phylogenetic trees that reflect viral evolutionary relationships. These trees reveal that giant viruses diversified hundreds of millions to over 1 billion years ago during the early evolution of eukaryotes. Different families represent ancient lineages that have been co-evolving with their hosts ever since. Accessory genes, by contrast, show rampant horizontal transfer and frequent gain and loss. A gene present in one giant virus species may be absent in a closely related species, or present in distantly related species from different families, reflecting independent acquisition events. This modular evolution allows viruses to rapidly adapt to new hosts or environments by acquiring pre-adapted genes rather than evolving new functions from scratch. The ancient permafrost viruses provide direct evidence for the stability of this mosaic structure over deep time. Comparing 42,000-year-old and modern genomes shows that while specific accessory genes have changed, the overall genome architecture, the mixture of vertically inherited core genes and horizontally acquired accessory genes, remains constant. This suggests that the evolutionary strategy of giant viruses, relying on gene acquisition to expand capabilities, has been stable for at least tens of thousands of years and likely far longer. One of the most intriguing findings is evidence of long-term persistence of viral strains in permafrost environments. Some viral genotypes detected in samples spanning thousands of years show minimal genetic change, suggesting they represent viable populations that persisted in the permafrost ecosystem across millennia. This challenges the assumption that permafrost is a static graveyard. Instead, it may be a dynamic ecosystem where microorganisms, including viruses, maintain low levels of activity even under frozen conditions. Research on bacterial activity in permafrost supports this idea. Certain bacteria remain metabolically active at temperatures as low as -20 degrees Celsius, dividing once every few decades or centuries. If bacteria can sustain slow metabolism, their viruses could also persist, occasionally infecting new hosts and replicating at extremely slow rates. Over thousands of years, this would result in viral populations that evolve minimally while accumulating minor mutations detectable through genetic analysis. Another possibility is that viruses detected at multiple depths represent separate deposition events, with the same viral lineage being frozen at different times rather than persisting continuously. Distinguishing between these scenarios requires detailed genetic analysis and dating, but both indicate that permafrost viral communities are more complex than simple frozen snapshots. The evolutionary insights from permafrost viruses extend to understanding how viral diversity responds to environmental change. The climate transitions captured in ice cores show that viral communities track their hosts. When host communities shift due to changing temperature, moisture, or other factors, viral communities shift correspondingly. Some viruses go locally extinct as their hosts disappear. Others invade as new hosts arrive. And some adapt, modifying host range or other traits to persist through the transition. These dynamics are relevant to understanding how modern viral communities will respond to ongoing climate change. If historical patterns hold, we can expect viral communities in thawing permafrost regions to undergo rapid turnover as environmental conditions change and microbial communities reorganize. Novel virus-host interactions may emerge as ancient viruses encounter modern hosts, potentially leading to new ecological dynamics or disease emergence. The frozen genomes in permafrost are not just historical curiosities. They are active participants in an evolutionary experiment spanning millennia. By studying these genomes, we learn how evolution operates over timescales inaccessible through laboratory experiments or observations of modern populations. We see the long-term consequences of natural selection, the persistence of stable genetic structures across environmental upheavals, and the fluidity of viral genomes that acquire and shed genes in response to ecological opportunity. This knowledge is foundational for predicting future viral evolution. If we understand the mechanisms by which viruses adapt to changing environments, hosts, and competitors, we can better anticipate how emerging viruses will evolve and how ecosystems will respond to viral invasions from thawing permafrost. The evolutionary history, written in frozen genomes, is not just a record of the past. It is a guide to the future, revealing the rules that govern viral diversity and adaptation on a warming planet. Chapter 12: The Virocell Concept and Viral Complexity. For over a century, viruses have been defined by what they are not. They are not cells. They do not metabolize. They cannot reproduce independently. Outside a host, a virus is an inert particle, a package of genetic information wrapped in protein. This definition, established in the early 20th century when viruses were first discovered, has shaped how scientists think about viral biology, evolution, and the nature of life itself. But the discovery of giant viruses has challenged this definition so profoundly that a paradigm shift is underway, reconceptualizing what viruses are and what it means to be alive. The traditional view holds that viruses exist in two states. Outside cells, they exist as virions, particles consisting of a genome enclosed in a protein capsid. Virions are metabolically inert. They drift passively through the environment, incapable of movement, growth, or response to stimuli. By most definitions of life, virions are not alive. They are complex molecular assemblies, but no more alive than a crystal or a machine. The second state is intracellular. When a virion encounters an appropriate host cell, it attaches to the cell surface, penetrates the membrane, and releases its genome into the cell interior. The viral genome then hijacks the cell's machinery, directing it to produce viral components. The cell becomes a factory for manufacturing new virions. Once enough components accumulate, they assemble into progeny virions that burst from the cell, killing it, and go on to infect new cells. During this intracellular phase, the virus is active, replicating, evolving, and exerting selective pressure on its host. But which entity is the virus? Is it the virion, the particle, or is it the infected cell, the replication factory? Traditional virology focuses on the virion, treating it as the virus and the infected cell as merely a hijacked environment. But this perspective becomes problematic when considering giant viruses, which blur the line between viruses and cellular life. The Virocell concept, proposed by Patrick Forterra and others, offers an alternative framework. It suggests that the infected cell is the actual viral organism, a new type of cellular life form distinct from both the uninfected host cell and the virion. The virion is not the virus itself, but a transmission stage, analogous to a spore or seed. Just as a plant seed is not the plant, but a dormant stage that grows into a plant under suitable conditions, a virion is a dormant stage that activates into a virocell upon infecting a host. This reconceptualization has profound implications. If the virocell is the organism, then viruses are cellular life forms, not acellular parasites. They are living entities during their intracellular phase, possessing metabolism through commandeered cellular machinery, reproducing by directing synthesis of viral components, and responding to environmental cues by regulating gene expression. The virocell is alive by most reasonable definitions, even if the virion is not. Giant viruses provide the clearest evidence for the virocell concept. When mimivirus infects an amoeba, it establishes a massive viral factory within the cell's cytoplasm. This factory is visible under a light microscope as a distinct compartment, up to several micrometers across, segregated from the rest of the cell. The factory contains high concentrations of viral DNA, transcription machinery, translation components, and assembling virions. It functions as a semi-autonomous unit, a proto-organelle within the cell. Recent research using advanced microscopy techniques reveals that these viral factories have internal structure. They are not simply bags of proteins and nucleic acids, but organized compartments with distinct zones. In some giant viruses, DNA replication occurs in a central region, surrounded by a zone where DNA is packaged into capsids, surrounded in turn by a region where capsids are assembled. This spatial organization resembles cellular organelles and suggests a level of biological complexity beyond what was thought possible for viruses. The virocell concept also helps explain the genetic complexity of giant viruses. Mimivirus encodes 979 proteins, including components of the translation apparatus like aminosyl RNA synthetases, translation initiation factors, and transfer RNAs. Why would a virus encode these components if it is merely hijacking the host cell's translation system? The answer becomes clear if we view the virocell as an organism that needs to control protein synthesis independently of the host. During infection, the virocell actively suppresses host gene expression while ramping up viral gene expression. The viral translation components may provide the virus with control over this process, allowing it to fine-tune protein synthesis according to viral needs rather than relying entirely on host machinery that is optimized for the host's needs, not the virus's. The virus is not just stealing the host's tools, it is bringing its own tools to modify the cellular environment to suit its replication strategy. Tupin virus takes this further. It encodes 20 aminosyl RNA synthetases covering all 20 standard amino acids, plus 70 transfer RNAs and multiple translation factors. This is the most complete translation apparatus ever found in a virus, approaching the complexity of some bacteria. While Tupin virus still depends on host ribosomes to actually synthesize proteins, it provides nearly everything else needed for translation. The virocell created by Tupin virus infection is almost a complete cellular organism, lacking only a few components to be fully autonomous. This raises the question of whether giant viruses represent an evolutionary transition towards cellular life. One hypothesis suggests that giant viruses are descendants of ancient cells that became parasitic and gradually lost genes no longer needed in the parasitic lifestyle. Over millions of years, these degenerate cells lost their ribosomes, their metabolic pathways, and other components, becoming streamlined parasites that retained only genes essential for replication and transmission. Under this scenario, giant viruses are reduced cells, and the virocell represents a partial recapitulation of their cellular ancestry. An alternative hypothesis proposes that giant viruses evolved from simpler viruses through gene acquisition. An ancestral virus, perhaps similar to a bacteriophage, infected early eukaryotic cells and began stealing genes. Over time, it accumulated hundreds or thousands of genes, becoming progressively larger and more complex. The genes it acquired included translation components, metabolic enzymes, and regulatory systems that allowed it to exert greater control over the infected cell and replicate more efficiently. Current evidence supports a mixture of both processes. Phylogenetic analyses show that some giant virus genes are ancient and distinctly viral, suggesting an ancient viral ancestor. Other genes are clearly acquired from eukaryotes, bacteria, or other viruses through horizontal gene transfer, supporting the gene accumulation model. The truth is likely that giant viruses have a complex evolutionary history involving both an ancient viral lineage and extensive gene capture. The virocell concept also reframes the debate over whether viruses are alive. Under the traditional virion-centric view, viruses are not alive because virions lack metabolism and cannot reproduce independently. Under the virocell-centric view, viruses are alive during the intracellular phase because virocells exhibit all the hallmarks of life. They metabolize, reproduce, evolve, respond to stimuli, and maintain homeostasis within the infected cell environment. This perspective aligns with how we think about other organisms with complex life cycles. Butterflies exist as eggs, larvae, pupae, and adults, with radically different forms and capabilities at each stage. We do not say butterflies are not alive during the pupal stage because pupae do not eat or move. We recognize the entire life cycle as a living organism passing through different stages. Similarly, viruses can be viewed as organisms with life cycles that include an inert transmission stage, the virion, and an active replication stage, the virocell. Giant viruses also challenge the three-domain model of life, which divides all cellular organisms into bacteria, archaea, and eukarya. If giant viruses represent a fourth lineage that diverged early in the history of life, they would constitute a fourth domain. This claim is controversial and not widely accepted, but it highlights how giant viruses do not fit neatly into existing classifications. One proposed solution is to recognize viruses as a separate category of life that evolves in parallel with cellular organisms, but through different mechanisms. Cellular organisms, termed ribosomes because they all use ribosomes for protein synthesis, evolve primarily through vertical inheritance with occasional horizontal gene transfer. Viruses, termed virosomes during their intracellular phase, evolve through rampant horizontal gene transfer, acquiring genes from any organism they infect and creating mosaic genomes that reflect their ecological promiscuity. The discovery that virophages can infect giant viruses adds another layer of complexity. If a giant virus is infected by a virophage, the virosome becomes host to a smaller virus, creating a nested hierarchy of parasitism. The virophage replicates within the viral factory, using resources the giant virus has concentrated there. This creates a three-level system: the amoeba cell, the giant virus virosome within it, and the virophage replicating within the viral factory. Each level has its own genome, its own evolutionary trajectory, and its own selective pressures. This nested structure challenges simplistic notions of what constitutes an organism. Is the virosome a single organism, or is it a composite of host cell, giant virus, and virophage, each pursuing its own agenda? The answer depends on how you define organism boundaries, a question that is not as clear-cut as it seems. In biology, organisms often consist of nested entities. Eukaryotic cells contain mitochondria, which are descended from bacteria and retain their own genomes. Humans contain trillions of bacterial cells in the microbiome, and these bacteria influence human health and behavior. The boundaries between self and other, organism and environment, are porous and context-dependent. The virosome concept embraces this complexity. It recognizes that during viral infection, the boundaries between virus and host dissolve. The infected cell is neither purely host nor purely virus. It is a hybrid entity with properties of both. The virosome has the virus's genome directing cellular activities, but it uses the host's ribosomes, membranes, and metabolic machinery. It is a transient biological entity that exists only during infection, ceases to exist when virions are released and the cell dies. But while it exists, it is as real and functional as any other cellular organism. For the ancient viruses emerging from permafrost, the virosome concept is particularly relevant. When a 48,000-year-old pithavirus infects a modern amoeba, it creates a virosome that bridges two eras. The virus's genes, shaped by selection during the Pleistocene, now direct a cell from the Anthropocene. The virosome is a genetic chimera, an organism that could not exist without both ancient virus and modern host, a living connection between past and present. Understanding viruses as virosomes rather than virions changes how we think about their ecological roles. Virosomes are metabolically active participants in ecosystems. They consume resources, produce waste, alter nutrient flows, and interact with other organisms in ways that virions cannot. A viral infection does not just kill a host cell, it transforms that cell into a different type of organism with different ecological functions. These transformations influence ecosystem processes at scales ranging from microbial communities in soil to oceanic nutrient cycles. The virosome concept also emphasizes that viruses are a source of evolutionary innovation. New genes can arise within virosomes through recombination, mutation, and interaction with host genomes. These viral genes can then be transferred to host genomes, where they may be co-opted for cellular functions. Much of the genetic diversity in cellular organisms originated in viruses and was transferred horizontally, making viruses not just parasites, but also engines of evolutionary creativity. As we study the 70,000 viral species hiding in permafrost, the virosome concept reminds us that these are not just frozen particles, but the dormant seeds of complex biological systems. When thawed and activated, they will create virosomes, transforming modern cells into hybrid organisms with ancient genetic programs. These viruses will interact with modern microbial communities, potentially influencing ecosystem functions in ways we cannot yet predict. The zombie viruses are not just relics of the past. They are potential participants in the ecosystems of the future, ready to resume their billion-year-old roles as parasites, predators, and evolutionary innovators in a world transformed by climate change. Chapter 13: Future Discoveries and Monitoring Systems. The race to document permafrost viral diversity is accelerating, driven by both scientific curiosity and urgent necessity. As Arctic temperatures continue climbing and permafrost thaws at unprecedented rates, the window for studying pristine frozen ecosystems is closing rapidly. Research teams across the globe are intensifying their efforts to collect samples, establish monitoring networks, and develop technologies capable of detecting emerging pathogens before they pose threats to human health or ecosystem stability. The future of viral discovery in the Arctic will be defined by international cooperation, technological innovation, and the recognition that what emerges from thawing ice affects the entire planet. Field expeditions to remote Arctic locations have become increasingly sophisticated operations. In 2025, multiple research teams conducted drilling operations across Siberia, Alaska, Greenland, and northern Canada, extracting permafrost cores from depths exceeding 100 meters. These expeditions face formidable logistical challenges. Equipment must withstand temperatures dropping to -40 degrees Celsius. Supplies must be transporting hundreds of kilometers across roadless tundra. Drilling sites must be selected based on geological surveys identifying regions where permafrost has remained continuously frozen for maximum time periods, often requiring helicopter access to isolated locations. The permafrost cores extracted during these expeditions represent vertical timelines extending back 50,000 years or more in the oldest deposits. Each core is carefully sectioned in the field, with samples taken at regular depth intervals. These sections are immediately frozen at -80 degrees Celsius using portable ultra-low temperature freezers powered by generators. Contamination control is paramount. All drilling equipment is sterilized before use. Personnel wear sterile gloves and face masks while handling samples. Each core section is wrapped in multiple layers of sterile material to prevent surface contamination during transport back to laboratories in Europe, North America, and Asia. The Permafrost Discovery Gateway, an international collaboration involving research institutions from the United States, Canada, Russia, Norway, and other Arctic nations, coordinates these sampling efforts. The gateway maintains a database of sampling locations, depth profiles, radiocarbon dates, and preliminary metagenomic data from each site. This allows researchers worldwide to access information about available samples and request subsamples for their own studies. The open access model ensures that permafrost research benefits the global scientific community rather than remaining siloed within individual laboratories or nations. Submarine permafrost, frozen ground beneath Arctic coastal waters that was exposed land during previous ice ages when sea levels were lower, represents another frontier for exploration. In fall 2025, the Monterey Bay Aquarium Research Institute conducted an expedition to study submarine permafrost dynamics in the Beaufort Sea north of Alaska. Using remotely operated vehicles equipped with sediment corers, the team collected samples from permafrost deposits 70 meters below the ocean surface. Preliminary analysis revealed abundant viral DNA, suggesting that submarine permafrost harbors distinct viral communities adapted to marine-influenced environments. These submerged permafrost deposits are thawing faster than terrestrial permafrost due to warming ocean waters, making their study particularly urgent. Next-generation sequencing technology continues advancing at remarkable speed, enabling ever more comprehensive characterization of viral diversity. The latest sequencing platforms can generate hundreds of billions of base pairs of sequence data in a single run, allowing complete reconstruction of viral genomes from environmental samples containing trace amounts of viral DNA. Portable sequencing devices like the Oxford Nanopore Minion, enable real-time sequencing in field conditions, providing preliminary results within hours of sample collection. Future Arctic expeditions may carry these portable sequencers, allowing researchers to identify high-priority samples while still in the field and adjust sampling strategies accordingly. Artificial intelligence is transforming viral discovery by enabling analysis of datasets too massive for human researchers to process manually. Machine learning algorithms trained on millions of known viral genomes can recognize viral sequences in metagenomic data, even when those sequences have minimal similarity to anything in existing databases. A study published in early 2026 described an AI system called VIRE, Virome Reference and Exploration, that analyzed planetary-scale metagenomic datasets and identified over 160,000 previously unknown viral species. Similar approaches applied to permafrost metagenomes could exponentially expand our catalog of ancient viral diversity. Deep learning models can also predict protein structures from genomic sequences, allowing functional characterization of unknown viral genes without laboratory experiments. When researchers encounter viral dark matter, genes with no similarity to known sequences, AI structure prediction tools can identify potential functional domains based on predicted three-dimensional protein shapes. This computational approach accelerates the process of assigning functions to hypothetical proteins and helps prioritize which genes warrant experimental investigation. Monitoring systems for emerging Arctic pathogens are beginning to take shape, though they remain underfunded relative to the scale of the challenge. The ideal monitoring network would include permanent research stations distributed across permafrost regions, equipped with automated environmental sampling devices that collect air, water, and soil samples on regular schedules. These samples would be analyzed for bacterial and viral pathogens using rapid molecular diagnostic tools, with results transmitted via satellite to central monitoring hubs. Several pilot projects are testing components of this vision. In Alaska, the Permafrost Pathogen Monitoring Network operates sampling stations at 5 locations across the North Slope. Each station includes automated water samplers that collect creek and pond samples weekly during the summer thaw season. Samples are shipped to the University of Alaska Fairbanks, where they undergo metagenomic sequencing to identify microbial communities, including potential pathogens. The system has been operational since 2023 and has successfully detected seasonal fluctuations in bacterial populations correlated with thawing depth and temperature. Similar monitoring efforts in Siberia focus on anthrax surveillance, given the 2016 outbreak that killed thousands of reindeer. Russian veterinary authorities maintain a network of soil sampling sites near historical anthrax burial grounds. During summer months, when permafrost thaws most extensively, samples are collected and tested for Bacillus anthracis spores. If spores are detected, alerts are issued to reindeer herders and local communities. Vaccination campaigns are initiated for livestock, and public health measures are implemented to prevent human exposure. Expanding these regional efforts into a comprehensive pan-Arctic pathogen surveillance network requires international coordination and sustained funding. The Arctic Council, an intergovernmental forum including Canada, Denmark, Finland, Iceland, Norway, Russia, Sweden, and the United States, has identified permafrost thaw and emerging infectious diseases as priority concerns. Working groups within the Council are developing frameworks for data sharing, standardized sampling protocols, and coordinated response plans for pathogen detection events. However, geopolitical tensions complicate cooperation, particularly regarding Russian Arctic territories where much of the most extensive permafrost exists. Indigenous Arctic communities are essential partners in monitoring efforts. These communities possess deep knowledge of local ecosystems, observe environmental changes firsthand, and are directly affected by permafrost thaw. Collaborative projects that combine traditional ecological knowledge with scientific monitoring, are proving highly effective. In northern Canada, Inuit community members work alongside researchers to collect environmental samples, report unusual wildlife die-offs or disease outbreaks, and provide historical context for observed changes. This partnership approach ensures that monitoring efforts address community priorities and that benefits from research flow back to the people living in affected regions. Climate modeling integrated with microbial ecology is beginning to predict where and when permafrost pathogens are most likely to emerge. These models incorporate data on permafrost temperatures, thaw depths, historical pathogen presence, wildlife migration patterns, and human population distribution to generate risk maps, identifying hotspots requiring enhanced surveillance. A model developed for Siberia successfully predicted that the Yamal Peninsula would experience heightened anthrax risk during the hot summer of 2016, though the prediction was not acted upon in time to prevent the outbreak. Improved models and faster response mechanisms could prevent future such events. Looking forward, the field of permafrost virology will likely see several major developments over the next decade. First, comprehensive metagenomic surveys will catalog the bulk of permafrost viral diversity, moving from the current 70,000 known species to perhaps millions as sampling coverage expands. Second, revival experiments will extend to older permafrost deposits, potentially recovering viable viruses from 100,000-year-old or even older ice. Third, functional characterization of dark matter genes will accelerate through AI-assisted prediction and high-throughput experimental validation, filling knowledge gaps about what ancient viral proteins do. Fourth, monitoring networks will mature from pilot projects into operational systems capable of detecting pathogen emergence in real time across the entire Arctic. Fifth, international cooperation will strengthen, driven by recognition that Arctic changes have global consequences and that no single nation can address these challenges alone. And sixth, ethical frameworks will develop around questions of whether certain ancient viruses should be revived, how samples from indigenous lands should be collected and studied, and who has authority to make decisions about pathogen research that could affect global public health. The future discoveries emerging from permafrost will reshape our understanding of viral evolution, diversity, and ecology. They will provide insights into how life adapts to extreme environments, how ecosystems respond to climate transitions, and how biological information persists across geological time. They will also confront us with practical challenges, monitoring vast, remote regions for emerging threats, responding to potential pathogen releases, managing the ecological consequences of introducing ancient microbes into modern ecosystems, and making ethical decisions about research that carries both tremendous scientific value and potential risks. The work happening now in Arctic research stations, in laboratories analyzing frozen cores, in supercomputers running AI algorithms on metagenomic datasets, and in international councils developing monitoring frameworks will determine whether humanity successfully navigates the Great Thaw. The zombie viruses are awakening, and our response to their emergence will test our scientific capabilities, our capacity for international cooperation, and our wisdom in balancing discovery with precaution in an era of unprecedented environmental change. Chapter 14: Implications for Astrobiology and Extraterrestrial Life. The discovery that viruses can survive in permafrost for 48,000 years has profound implications extending far beyond Earth. It demonstrates that complex biological molecules, including DNA genomes and functional proteins, can remain viable under frozen conditions for geological timescales. This finding directly informs the search for life on other worlds, particularly those with ice-dominated environments. If Earth viruses can persist intact through tens of thousands of years of frozen dormancy, similar preservation might occur on Mars, on the icy moons of Jupiter and Saturn, or in other frozen environments throughout the solar system and beyond. Mars is the most obvious application. The red planet possesses extensive ice deposits at its poles, subsurface ice across much of its mid and high latitudes, and a history of liquid water on its surface billions of years ago. If life ever existed on Mars during its warmer, wetter past, remnants of that life might be preserved in ice. The permafrost analog suggests that Martian ice could act as a biological archive, storing microorganisms or viruses that were active when Mars had lakes, rivers, and potentially habitable conditions. Recent studies directly tested this hypothesis using Mars analog experiments. Researchers from NASA Goddard Space Flight Center and Penn State University exposed bacteria to simulated Martian conditions, including extreme cold, low pressure, and continuous exposure to cosmic radiation similar to what Mars experiences. The bacteria were embedded in water ice and irradiated with doses equivalent to millions of years of Martian surface exposure. The results, published in 2025, showed that amino acids from the bacterial proteins survived with more than 10% remaining intact after simulated exposure equivalent to 50 million years on Mars. This survival rate dramatically exceeds previous estimates. Earlier models predicted that cosmic radiation would destroy biological molecules within a few million years at most. The new findings suggest that in pure ice, radiation damage proceeds much more slowly than in ice mixed with soil or other materials. The ice matrix apparently traps reactive particles created by radiation, preventing them from reaching and damaging organic molecules. This protective effect means that Martian ice deposits could preserve signatures of ancient life for geological timescales comparable to the age of the ice itself. The implications for Mars sample return missions and future crewed exploration are significant. If biological material survives best in pure ice or ice-dominated permafrost, future missions should target these environments rather than focusing exclusively on sedimentary rocks or clay deposits. The Mars Phoenix lander in 2008 confirmed the presence of water ice just beneath the Martian surface in the Arctic region. Subsequent missions, including the Mars Reconnaissance Orbiter, have identified extensive subsurface ice deposits across the mid-latitudes. These ice deposits, some of which may be hundreds of millions of years old, are prime targets for life detection. A mission concept currently under development envisions a robotic lander equipped with a drill capable of penetrating several meters into Martian permafrost to extract ice cores. The cores would be analyzed in situ using miniaturized sequencing technology to search for biomolecules including amino acids, nucleic acids, and lipids. If signatures of life were detected, a follow-up sample return mission could bring Martian ice back to Earth for comprehensive analysis in terrestrial laboratories, following strict planetary protection protocols to prevent contamination in either direction. The technological parallels between permafrost viral research on Earth and astrobiology are striking. The same metagenomic sequencing methods used to identify viruses in Siberian permafrost could detect Martian microorganisms in ice samples. The same contamination control protocols ensuring that terrestrial viruses do not compromise permafrost samples apply to preventing Earth life from contaminating Mars samples and Martian material from contaminating Earth. The scientific questions are analogous. What biological diversity existed in ancient frozen environments? How long can life survive in ice? What factors determine preservation versus degradation? Europa, a moon of Jupiter, presents another compelling target. Beneath its icy crust lies a global ocean of liquid water, maintained in liquid state by tidal heating from Jupiter's immense gravity. The ocean is estimated to contain more water than all of Earth's oceans combined. Plumes of water vapor erupting from Europa's surface, detected by the Hubble Space Telescope and confirmed by later observations, suggest the subsurface ocean connects to the surface through cracks in the ice. If life exists in Europa's ocean, organisms or viruses could potentially be transporting to the surface in these plumes and frozen into the ice crust. Same radiation survival experiments conducted for Mars apply to Europa, though with different parameters. Europa's surface experiences intense radiation bombardment from Jupiter's magnetosphere, far more severe than cosmic radiation on Mars. However, research published in 2024 demonstrated that amino acids and other biomolecules could survive near Europa's surface at high latitudes for timescales of millions of years if buried just 20 centimeters beneath the ice. The ice provides shielding from radiation, and the extreme cold preserves molecular structure. Future missions to Europa should therefore sample the near-subsurface ice rather than just surface material. NASA's Europa Clipper mission, launched in 2024, will conduct detailed reconnaissance of Europa during multiple flybys beginning in the early 2030s. While Clipper is not equipped to land or drill, it will map the surface in unprecedented detail, identify optimal landing sites, and potentially fly through erupting plumes to sample material directly ejected from the subsurface ocean. A follow-up lander mission, currently in conceptual development, would touch down on Europa's surface, deploy a drill or heated probe to access subsurface ice, and analyze samples for biosignatures. Enceladus, a moon of Saturn, also erupts plumes of water from a subsurface ocean. The Cassini spacecraft flew through these plumes multiple times between 2005 and its mission end in 2017, detecting organic molecules including complex carbon compounds. The presence of these organics, combined with evidence for hydrothermal vents on Enceladus's ocean floor, makes the Moon one of the most promising locations in the solar system for finding life. Ice particles from the plumes fall back to Enceladus's surface, creating fresh ice deposits that could contain frozen microorganisms if they exist in the ocean. The European Space Agency has selected a mission concept for development targeting Enceladus. The proposed mission would include both an orbiter to study the Moon from space and a lander to touch down on the surface, collect samples from fresh plume deposits, and analyze them for signs of life. The technological challenges are immense, requiring spacecraft to survive the journey to Saturn, navigate into orbit around Enceladus, and successfully land on a distant, barely known world. But the scientific payoff could be correspondingly enormous: the first confirmed detection of extraterrestrial life. The permafrost virus research provides a template for how to handle samples from these icy moons. If samples were returned to Earth, they would need to be kept frozen continuously to preserve any microorganisms or viruses they might contain. Analysis would proceed through a combination of sequencing, microscopy, and culture attempts, exactly paralleling the methods used for permafrost samples. If viable extraterrestrial microbes were detected, revival experiments analogous to Clavery's zombie virus work would be conducted under the highest biosafety containment to ensure Earth's biosphere is not contaminated with alien organisms. The concept of panspermia, the hypothesis that life can spread between worlds through space, gains plausibility from permafrost virus survival. If viruses can remain viable for 50,000 years in Earth ice, could they survive the journey through space embedded in meteorites ejected from one planet and landing on another? Mars meteorites have reached Earth, blasted from the Martian surface by asteroid impacts and drifting through space before eventually falling to our planet. Some of these meteorites contain ice or minerals formed in the presence of water. If Martian microorganisms existed, could they hitchhike to Earth in such meteorites? The challenges are formidable. Meteorites ejected from planetary surfaces experience extreme acceleration, then passage through space where they are exposed to vacuum, radiation, and temperature extremes, then fiery atmospheric entry heating them to thousands of degrees. Most scientists consider it unlikely that life could survive this sequence. However, if organisms were embedded deep within a large meteorite, the interior might remain cold and shielded throughout the journey. Experiments testing bacterial spore survival under simulated meteorite impact and atmospheric entry conditions have shown that some spores survive in protected locations within rock samples. More plausibly, dormant life could spread between icy moons in the same planetary system. Europa and Enceladus occasionally experience meteorite impacts powerful enough to eject material into space. If those impacts excavate subsurface ice containing microorganisms, The microbes could be blasted into orbit and eventually fall onto neighboring moons. In the cold of space, frozen in ice fragments, they might survive the journey and seed life on a new world. The timescales involved could be millions of years, but if permafrost viruses survive 50,000 years on Earth, extremophile organisms in space ice might persist for comparable or longer durations. The discovery of viable ancient viruses also informs the search for life on rogue planets, worlds that have been ejected from their star systems and drift through interstellar space. Without a star to provide heat, these planets would be frozen solid, but internal heat from radioactive decay could maintain subsurface liquid water, creating potential habitats for life beneath ice crusts kilometers thick. If life emerged on such worlds during their early history when they still orbited stars, it might survive in the subsurface for billions of years. The ice above would preserve any organisms that became frozen, creating time capsules of ancient ecosystems. Even on Earth, the permafrost viral research has implications for theories about life's origins. The earliest evidence for life on our planet dates to approximately 3.8 billion years ago, but Earth experienced repeated global glaciation events, including snowball Earth episodes when the entire planet may have been encased in ice. How did life survive these frozen periods? The permafrost analog suggests that even if surface oceans froze solid, microorganisms frozen in ice could remain viable for millions of years until the planet warmed and ice melted, allowing life to resume. The search for biosignatures on exoplanets orbiting distant stars can also learn from permafrost research. Telescopes capable of analyzing the atmospheric composition of rocky exoplanets are beginning to come online. If such planets show evidence of oxygen, methane, or other potential biosignature gases, follow-up observations could search for seasonal variations that might indicate biological activity. But if the planet is frozen, life might be dormant during most of its orbit, becoming active only during brief warm periods. Understanding how life behaves in frozen environments helps astrobiologists predict what signatures to look for on cold worlds. The 70,000 viral species in permafrost represent the largest collection of viable ancient life on Earth. They are evolutionary time capsules, genetic archives, and functional organisms frozen in time. They prove that complex biological systems can survive not just years or centuries, but tens of thousands of years under the right conditions. This knowledge fundamentally changes how we search for life in the solar system and beyond. Ice is not a grave, but a library, preserving information about past life and potentially maintaining viable organisms across geological timescales. As we drill into Martian permafrost, sample Europa's ice, and analyze plume particles from Enceladus, we will apply lessons learned from Earth's zombie viruses. The implications extend from the Arctic tundra to the outer solar system, from ancient Earth ecosystems to potential alien biospheres, from the limits of terrestrial life to the possibilities of life throughout the universe. Chapter 15: Living in the Age of Awakening. We stand at a moment unprecedented in planetary history. For the first time in 50,000 years, vast regions of Arctic permafrost that have remained continuously frozen since the Pleistocene are thawing. And for the first time ever, humanity possesses the scientific tools to study what emerges from that ancient ice. These two firsts converge to create a unique opportunity and responsibility. The frozen time capsule is opening, revealing a hidden world of microbial diversity that includes an estimated 70,000 viral species, countless bacterial lineages, and genetic information from ecosystems that vanished millennia ago. What we do with this knowledge will shape our relationship with a rapidly changing planet. The paradox of our moment is stark. Climate change, driven primarily by human activities, threatens ecosystems worldwide, destabilizes Earth's climate system, and poses existential risks to human civilization. Yet this same climate change is revealing the permafrost archive, allowing us to study ancient viruses, trace evolutionary histories across deep time, and gain insights into how life adapts to environmental upheaval. We are destroying the frozen library even as we learn to read it. Every summer, more permafrost thaws. Every year, opportunities to study pristine ancient ecosystems diminish. The race between scientific discovery and environmental destruction defines the era we live in. The 70,000 viral species hiding in permafrost represent just a fraction of total viral diversity on Earth, which likely numbers in the tens of millions of species. Yet these particular 70,000 are special because they are frozen snapshots from the past. They show us what viral communities looked like during ice ages when mammoths and saber-toothed cats roamed the Arctic. They preserve genetic sequences from extinct viral lineages that evolved solutions to ecological challenges we can study and potentially learn from. They demonstrate the resilience of biological information, surviving conditions that would destroy most life. These ancient viruses also remind us of the fragility of ecosystems. The organisms they infected, the ecological networks they participated in, the environmental conditions they thrived under, all have vanished. The modern Arctic bears little resemblance to the Pleistocene tundra steppe ecosystem that existed 48,000 years ago. The megafauna are extinct. The climate is different. The plant communities have changed. Yet the viruses persist in ice, ready to awaken when thawed, searching for hosts that may no longer exist or encountering modern hosts with which they have no evolutionary history. What happens when ancient viruses meet modern ecosystems is unpredictable. Most thawed viruses will likely find no compatible hosts and simply degrade in the environment. Some will encounter distant relatives of their ancient hosts and may or may not successfully infect them. A few might infect modern organisms, potentially influencing ecosystem processes in subtle ways. The probability of a catastrophic pandemic from permafrost viruses remains low, but monitoring and preparedness are essential because low probability is not zero probability, and the consequences of an outbreak from an unknown ancient pathogen could be severe. Our responsibility in this age of awakening is multifaceted. First, we must document every permafrost core extracted, every metagenomic sequence generated, every viral genome assembled adds to our catalog of ancient diversity. This documentation creates a permanent record that will outlast the permafrost itself. Future generations will study these sequences long after the ice has melted, learning from genetic information that would otherwise be lost forever. Second, we must monitor. Surveillance systems for emerging Arctic pathogens, while currently limited, must be expanded and sustained. We need networks of environmental sensors, rapid response teams capable of investigating unusual disease events, and international cooperation frameworks that allow data sharing across borders. The 2016 anthrax outbreak in Siberia demonstrated what can happen when permafrost pathogens are released. Better monitoring might have detected the outbreak earlier and prevented some deaths. Third, we must understand the virocell concept, viral dark matter, evolutionary dynamics visible in permafrost genomes. These are not abstract academic questions, but foundational knowledge needed to predict how ecosystems will respond to ongoing environmental change. Understanding how ancient viral communities shifted during past climate transitions helps us anticipate how modern communities will respond as temperatures continue rising. Fourth, we must act on climate. Slowing Arctic warming reduces the rate of permafrost thaw, preserving more of the frozen archive for future study and reducing the risks of pathogen release and massive carbon emissions. Every fraction of a degree of warming prevented matters. The difference between 2 degrees Celsius and 3 degrees of global warming translates to hundreds of thousands of square kilometers of permafrost preserved versus thawed, millions of tons of carbon released versus sequestered, and countless species lost versus maintained. Fifth, we must communicate. The public understanding of zombie viruses is often shaped by sensationalized media coverage, emphasizing pandemic scenarios while downplaying scientific nuance. Effective science communication that conveys both the genuine risks and the fascinating discoveries emerging from permafrost research is essential. People need to understand that ancient viruses are not monsters from horror movies, but real biological entities with implications for ecology, evolution, and public health. Sixth, we must engage ethically. Questions about whether certain ancient viruses should be revived, who has authority over permafrost samples collected from indigenous lands, how the benefits and risks of this research should be distributed. These require thoughtful deliberation involving diverse perspectives, including scientists, ethicists, policymakers, and Arctic communities whose homelands are the source of these samples. The age of awakening we live in extends beyond viruses to the broader transformation of the Arctic. The region is experiencing changes that would have been unimaginable a few decades ago. Sea ice is disappearing, glaciers are retreating, ecosystems are reorganizing as species shift ranges northward. Indigenous communities are adapting to landscapes that no longer follow the seasonal patterns their ancestors knew for thousands of years. The entire Arctic system is in flux, transitioning from the frozen state that characterized it for most of human history to something new and uncertain. Within this transformation, the zombie viruses are one thread in a complex web. They are not the primary threat, which remains the climate disruption itself. They are not the most important discovery, which might be the permafrost carbon feedback amplifying global warming. But they are emblematic of the surprises awaiting us as we continue altering Earth's climate. We are conducting a planetary-scale experiment with outcomes we cannot fully predict. Ancient viruses emerging from ice are one consequence. There will be others, some we can anticipate, many we cannot. The scientific community studying permafrost viruses operates with the knowledge that their work exists at the intersection of discovery and urgency. Every research expedition might find something extraordinary, a virus unlike any other, a genomic sequence revealing unexpected evolutionary pathways, evidence of extinct viral families that could reshape our understanding of viral diversity. But every expedition also confronts the reality that the subjects of study are disappearing. Permafrost thaw is accelerating, the archive is burning. If there is hope, it lies in our capacity for ingenuity, cooperation, and wisdom. Humanity has shown it can respond to global challenges when sufficiently motivated. The discovery of the ozone hole led to international agreements phasing out ozone-depleting chemicals, allowing the ozone layer to recover. The threat of nuclear annihilation during the Cold War motivated arms control agreements that reduced arsenals. Climate change is a more diffuse challenge with no simple technological fix, but the fundamental tools exist: renewable energy, sustainable agriculture, ecosystem restoration, carbon sequestration. What we need is the collective will to deploy these tools at the scale and speed required. The zombie viruses will continue awakening as long as permafrost continues thawing. We cannot prevent this entirely. Some warming is already locked in by past emissions, but we can slow it, manage it, and prepare for it. We can build the monitoring systems that detect emerging pathogens early. We can conduct the research that reveals which ancient viruses pose risks and which are harmless. We can develop the response capabilities to contain outbreaks if they occur. We can integrate knowledge from permafrost research into broader strategies for managing ecosystem changes in a warming world. The 70,000 species hiding in plain sight are not hiding anymore. They are emerging, documented in metagenomic databases, revived in laboratories, studied in exquisite detail. They tell us stories about the past, about Pleistocene ecosystems and the evolution of life across ice ages. They inform our understanding of the present, revealing how viruses survive extreme conditions and how ecosystems respond to climate shifts. And they warn us about the future, showing that our actions today will echo through time, releasing biological entities that have been frozen since before human civilization began. We live in the age of awakening, when ancient viruses stir from frozen sleep, and when humanity awakens to the consequences of planetary-scale environmental change. How we respond will determine not just what we learn about these extraordinary frozen libraries, but whether we preserve what remains, whether we navigate the Great Thaw safely, and whether we leave a stable planet for the generations who will continue this work after us. The zombie viruses are rising, And with them rise questions of science, ethics, and survival that define our relationship with a changing Earth. If you found this exploration of zombie viruses and the hidden world within permafrost compelling, hit that like button and subscribe for more deep dives into the intersection of science, climate, and the unexpected discoveries reshaping our understanding of life on Earth and beyond. The frozen frontiers are thawing, the ancient secrets are emerging, and the story is just beginning.